哎，彭教授好，听得见吗？彭教授，
你好，你好，你好，你好，
听得见是吧？
对，听得到我们的，
你那边声音我听着不清楚，我这边你听得清楚吗？
这个你知道吧？
哎，听到了，听到了。好，
Sound check.
行，那就彭教授准备好了吗？
Okay, so let's start.
好，
Okay,
大家好，
good
我
afternoon.
是国家食品安全风险评估中心的贾
I'm Gao Xudong from the China National Center for Food Safety Risk Assessment. It is my great honor to share with Mr. Peng this breakout session. Due to the pandemic, I cannot come on site, so I can only monitor online. For this afternoon, we have invited experts from academic and research institutes, etc., to talk about the non-animal methods for safety testing, NAMs. There is another meaning for NAM. And I believe that the presentations will be related with this topic. And if you're interested, you can also raise questions and discuss with the experts. And we have simultaneous interpretation, so you can feel free to use either Mandarin or English. So the first presentation is strategy for application of new approach methodologies in food safety assessment. And the speaker is Tang Dawei, the research scientist from the UK, and he has prepared a video. And if you have any question, he will be able. He is available online later. So, Mr. Tang. Dr. Tang joined Unilever in 2011 after he obtained his PhD in decision science and operational research from the University of Manchester in the UK, and is currently a research scientist in the Safety and Environmental Assurance Center of Unilever based in Covers R&D. And he has three current roles: first, applying a PPK modeling, and second is eliciting expert knowledge and developing mathematical models, and third is advocating next generation food safety risk assessment method in China. Next, please play the video.
Hello everyone, my name is Dawei Tang. I come from
有没有声音了哈？
我这什么都听不到了。
可能现场的问题，可能在调试呢。
Hello everyone, my name is Dawei.
看着现场提前没有调试啊。大家稍微等一下啊，可能出了一点技术问题。
贾教授，
哎，
Professor Jia, there is something wrong with the video playing. So now let's go to the second speaker, okay? Okay, Mr. Peng, the floor is yours.
考虑考虑，我们不行的话，我们就现场讲了哈。好，好，请彭教授主持第二个吧。
Okay, the second speaker is from East China University of Science and Technology, Professor Tang Yun, and his topic is chemical safety assessment via computational toxicology. So I'd like to briefly introduce Professor Tang. So in 1996, he received a PhD degree from Shanghai Institute of Materia Medica, Chinese Academy of Sciences. From 1996 to 2004, he studied and worked in Sweden, the US, and Canada successively. In 2004, he returned to China as a professor at Fudan University. In the same year, he participated in the establishment and discipline construction of the School of Pharmacy at East China University of Science and Technology. Since 2006, he has worked at ECUST full-time. He is mainly engaged in computer-aided drug design, chemoinformatics, computational toxicology, etc. He has published more than 230 SCI indexed papers in professional journals and obtained 7 computer software copyrights. Next, the floor is yours, Professor Tang. Okay. Thank you, Mr. Professor Peng, for the introduction, and thank you for giving me this opportunity to present the chemical safety assessment via computational toxicology. We all know that in our lives, there are various kinds of chemicals, and now there are more than 150,000 kinds of chemicals closely related to people's daily life, such as medicine, pesticides, food ingredients, and additives, cosmetics, industrial chemicals, etc. These chemicals not only improve people's quality of life, but also bring potential human health problems and environmental risks. So that's why the risk assessment, chemical safety assessment is very important. But before entering the market, those chemicals have not gone through very robust toxicology test. Only drugs have gone through very robust toxicity test. But actually, that robust test was not there from the very beginning. It was only getting strict after certain incidents happened. But the traditional toxicology test uses animal study, but today our topic is non-animal methods, and we know that the animal test has many problems. So that's why there are many development of non-animal methods. And computational toxicology is a very important non-animal method. Computational toxicology is also called predictive toxicology. It is a cross-discipline. It includes 3 characteristics in my understanding. So first is the construction of prediction models based on existing experimental data. AI methods are used to construct various toxicity prediction models, including qualitative classification, etc. And second is identification of structural alerts to identify the structural alerts of various toxic endpoints from massive data using AI methods. So these are preferred by chemists. So they look at the structure to see whether there is toxicity. And it's also easier for structural modification. And the third is computational systems toxicology. It can clarify the mechanisms of chemical toxicity so as to better understand how the toxicity comes from and how to avoid the toxicity. So the final goal is 3R, which we are very familiar with. Which is to replace laboratory animals, to reduce the number of lab animals, and to refine animal experimental procedures. And I'd like to briefly talk about AI, artificial intelligence. Artificial intelligence refers to allowing machines to do things that require human intelligence, such as learning, reasoning, and decision-making. It is a data-driven computer program. So there are three elements of AI: data, algorithms, and computational power. So in the past, we only had algorithms. So that's why AI actually was not so widely applied. But with big data, AI is closely combined with big data. Because big data offer a lot of learning materials to AI, and then it started to be widely applied. And actually, AlphaGo in 2016 won the chess campaign. Then after that, AI has been accepted more and more. AlphaGo uses deep learning algorithms. Now AI is widely used in a variety of fields, such as chemical risk assessment and management. Now, in the coming era of big data and AI, computational toxicology has entered a new stage, which can make full use of the massive toxicity-related data and further improve the prediction accuracy of toxicity prediction models. And it's very helpful. Now the US and the EU have advanced in computational toxicology and have related legislation. And some computational toxicology can be used to replace animal tests. And in EU cosmetics, some have banned the use of animal tests. Well, in China is also following up quickly in recent years. So this lists the projects, the databases online, and also including business databases, free databases, as well as some predictive tools, some commercial and also free tools. So this is a tool developed by our lab, which I will describe later. Our lab has been focusing on this area in recent years, and we have made some achievements in all three of the aspects. We have established a series of prediction models, and we also have the online prediction systems, and we have those publications. And we have also published some methodology, and we also have publications in computational systems toxicology. In the interest of time today, I will mainly talk about the model building for in silico toxicity prediction, which is easier to understand. So the toxicity prediction model mainly uses chemical category approach. It's like the traffic lights. For example, red means that chemical is toxic, and yellow means this chemical may be toxic or has low toxicity, and you need to be aware of. And the green means no toxicity. And we have used classification model, including binary, multi-class, and multi-label classifications. And OECD has, this is the OECD principles for QSAM models. There are 5 principles for QSAM model. First, defined endpoint. Say which toxicity you want to study, and there needs to be an unambiguous algorithm, a defined domain of applicability, and the fourth is appropriate measure of goodness of fit, robustness, and predictivity, and the fifth is a mechanistic interpretation if possible. This is a procedure of model building. First, it starts with data collection and then data description, feature selection, model building, model evaluation, and finally is the definition of applicability domain, altogether 6 procedures.
I would like to brief about the data sources, and we have some known databases that have been widely used. Now we can collect data from such databases. And we also have some data scattered in different literatures, and we shouldn't only focus on the database. And sometimes the data isn't updated timely. And next, more molecular representation and how we can make the computer to recognize the molecular structure. And in the past, and we got some parameters to describe the molecules, but it is only to transfer the molecular structures into the data. There was some deviation. However, now we have molecular fingerprints. And for example, we pre-define structure and the positions, and on each position there is a sub structure. And if on this position there is a sub structure, it is marked with 1, and without marked with 0. And so the molecular fingerprints and can be marked, and we can see if for this molecule, and the 1 is for 4 positions, and then it would be converted into the binary codes, and very easily to be dealt with by the computer. And another way is the graph neural networks, because if we adopt the graph neural nets to describe the molecule, it is the most straightforward way. And there are mainly 3 types of prediction. And the first is the classification models, which can be divided into classified into red, yellow, and green. And then we use the deep learning machine learning methodology to make it. And if we want to get a specific value of a toxicity, sometimes if we don't know what it presents for, and so the classification model can help us to resolve that issue, and it is commonly used. And if we can only follow some similar structured molecules, and we know the nature of such molecules, and then we can learn from this structure and to read across about the predicted molecule. And so we can see the model validation for classification models, and we need statistical data, true positives, and means if it is toxic and you predict it as toxic, then it is a TP. And but if it is toxic, but you predict it to be non-toxic, then it is a false negatives FM. And so the sensitivity, specificity, as well as the overall accuracy can all be used to describe. And we also figured out a way and to identify the substructures, and it is called information entropy, and to identify which molecules are toxic. I will give you an example. What kind of substructures are related to the oral acute toxicity. And so we can see the models are related to the chemical arms, mutagenicity, acute oral toxicity, endocrine disruption, carcinogenicity, liver toxicity, etc., as well as the respiratory toxicity. We have built such models, and give you an example about in silico prediction of a chemical acute oral toxicity. And we adopted the EPA's criteria, and so there are 4 categories demonstrating the toxicity level, and we collect the data, and totally more than 10,000 diverse compounds. And we also have external validation sets, and there are 2 fingerprints we used. MACOS and FP4. And here is the statistics of data sets. And because we categorize the compounds into 4 classifications: extremely toxic, highly toxic, moderately toxic, and slightly toxic. And so we totally classified over 12,000 compounds, and we can see from extremely toxic to the slightly toxic, and we have listed 4 categories here. And we have adopted 5 machine learning methods, as well as the descriptions of molecules, and we built 12 classified models. And we can see the performance of models for test set and which structure is better. And one is the Q value, and the other is the PPV value. And we can leverage such values to judge which prediction is more accurate. And we can let's look at the results. And the best model, the accuracy is close to 70 to or 80%. And so the closer to 1, the better. And so these 2 models are better in performance for a test set. But however, the samples were only over 10,000. So the accuracy can only be around 80%, but very hard to reach up to 90%. And then let's look at the external validation set, and the prediction accuracy is about 60 to 70%. Is not related to the modeling, but we can see this data set is better with the accuracy around 90%. And we use our methodologies and to identify the substructures in category 1. And we can see this is the extremely toxic substructures, and also some compounds containing such substructures. If there are such substructures in the molecules, it means that it is extremely toxic. So we need to avoid such chemicals in the food additives. And the second category of substructures are shown like this. And it's just a simple example. But in addition to building some models and to serve the public, and we also established a web server development that can be used by the math public. And here is the website. If you are interested, welcome to try it. And we collected almost 100,000 compounds with 210,000 pieces of data values, and we set up 22 qualitative classification models, and as well as 5 quantitative regression models. And here are some toxicity prediction types, including the drug-induced toxicity, acute toxicity, AMIS mutagenicity, etc. And we published a paper in November 2012. 10 years has passed, and it has been quoted or cited for more than 1,000 times. And drug banker and started to use our system since 2014 to predict the ultimate properties of drugs, and including the drug kinetics, as well as the toxicity. And the paper was selected as one of the 10 most influential papers of the PHID journal so far. On the 60th anniversary of JCIM in January 2021. And our paper was selected. And back in 2019, and our system has been updated to the 2.0 version. And we can see there is a tool in the website, and we set up more models, around 40 binary classification models, 3 multiple classification models, and 4 regression models, because we have new data to remodel, and so the model can be more accurate. And if we found some substructure is not very nice, and we can substitute, replace it. And this paper has been cited, and in the past 3 years, it has been cited by almost 400 times so far. And we are ready to update the system to 3.0 version. And currently, we are working on more than 60 models, and the classification will be better. And I would like to brief about the use of this system. And when you enter the system, this is the interface, and you can draw a molecule, the structure of the molecule, and it will be converted into a smiles, and then you click the predict, and it can go through prediction. And when the prediction is done, and the result can be shown immediately. And for example, this is the BPA, and you can check the physical and the chemical properties. And for example, this is the human intestinal absorption, FACO2, blood-brain barrier, and maybe you can't see it clearly. And if there is a plus symbol, and it means that it is a positive compound, it can be absorbed by the intestine, human intestine. And the probability represents that that almost 99% it can be absorbed by the human intestine. And here are some properties directly related to the metabolism. And then the toxicity, different kind of toxicity, and we can see the carcinogenicity, and eye corrosion, eye irritation, and Ames metagenesis, and respiratory toxicity, reproductive toxicity, mitochondrial toxicity, etc. And there are some quantitative prediction. And for example, the water solubility, plasma protein binding. Okay, the time is limited, and I just give you a summary. And this is the firstly, and we developed some methods. We adopted molecular fingerprint, machine learning, information gain, web server, activated SAR, and we also adopted the deep learning method to establish the model. And the information gain is used to identify the key substructures in molecules. And we established the web server Admit SAR, which is quite known internationally. And we also have some applications for chemical acute oral toxicity, Ames metagenicity. Okay, that's all from me. Thank you. Thank you, Professor Tang, for your wonderful presentation. And he has briefed about what they have done in the past 10 years in terms of the computational toxicology. And he showed us their case studies as well as the website service. Any question from the audience? And thank you for your wonderful presentation. And I have several questions. The first question, and I noticed that the models are mostly based on over 10,000 compounds. And my first question is that if some compounds and are in similar structures, and then maybe there are 300 compounds, and if the over 10,000 compounds are in different structures, whether the accuracy is higher. For example, there are 300 compounds of the same of the similar category, then you can establish the quantitative models. And so quantitative model is more accurate, and but it's only for this type of structures, but it's not very accurate to other structures. And then, yes, I had a question that for more than 10,000 compounds, and if it is a molecular fingerprint, and if it is the variance 0, 1, and then the outcome is also 0, 1. And there would be some information loss in this process, but you mentioned that this method can reduce the deviation. And so, and another advantage is it is more suitable for extrapolation. Extrapolation, right? Yes, it can be widely used.
So the classification accuracy is only around 60 to 70%. It can be also as high as 90%. Yes. So if it is a quantitative indicator and not acute toxicity, for example, just a distribution coefficient, if I use a quantitative coefficient, how many models I need to achieve a relatively high accurate prediction? Actually, there is no standard here. But for example, dozens of compounds can also be sufficient to build a very good model. For example, like the quantitative analysis of drugs. The experiment result can still be very accurate, but the more compounds there are, in terms of machine learning, the more information the machine can learn, right? Particularly both the positive and negative information. Some are quite active, some are not that active. And there are different characteristics that should be learned. And as a result, the model's prediction result will be more accurate. Okay, thank you. Thank you again. Okay, the next speaker, next Professor Jia, please moderate the next presentation. Okay. So, Professor Tang, is it okay for you to share the screen and deliver the speech in real time?
能看到屏幕吗？
看到了。OK，你把它放大一下，全屏。
好，等一下啊。
好，可以了。好，下面请大卫博士做报告。
我还是用英文吧，因为在座还有很多国外的人士。Okay, so it's my pleasure to be invited to give this presentation on this conference. The title of my presentation is the strategy for application of new approach methodologies in food safety assessment. This presentation will have 5 parts. So after a general introduction, I will introduce NGRA framework. NGRA here is a new generation risk assessment. And I will introduce some methods of the NGRA, and I will use a sulforaphane risk assessment case study to demonstrate how this method has been or can be applied. And then there's a summary and the discussion part. So first, there's a very quick one is that I come from Unilever, and I think many people here know what Unilever is, and it's one of the world's biggest consumer good companies with over 400 brands. And I'm sure that many of you can recognize many of those brands. And then we have like over 400 brands, and 13 of the top 50 consumer goods brands come from Unilever. So the question is that why a consumer goods company is so interested in the risk assessment area, because there is a department called Safety and Environmental Assurance Center, which is CIFSQ, this department in Unilever is specifically responsible for the product safety of Unilever. So this is where I come from. That's why we are interested in this risk assessment of consumer goods. So CIFSQ is industry leading safety and environmental sustainability science capability, and we developing science, we shared science, and also we applied science. And I think the purpose of our I would say one of the purpose of us is to leverage our science and global networks for consumer trust and freedom to operate. And also, we are also here to ensure that our Unilever products have the meet our internal ingredient safety governance requirements. So this is CIFSQ where I come from, and traditionally, by traditionally I mean many years ago, as Professor Tang just mentioned, that we do the risk assessment by animals, and basically we apply a dose of a chemical on a certain animal, and then we observe its no observed adverse effective level, and based on that level, we divide it by 100, in which 10 represents the difference between animal and human, and the other 10 represents the population variance. So we use this no-well level divided by 10, and we can get this. Wait a moment. We can get this ADI or TDI, and on the other hand, we estimate the exposure. If the exposure is less than the ADI or TDI derived from the animal study, we say it is safe. If the exposure is above this ADI or TDI derived from this no-well, we say it is not safe, or we say there's a risk. And this method has been applied in many years, but especially in the 21st century, then more and more people realize that there's a lot of limitations of this method, and one of them is that the value of animal test is being challenged, because animal is not human. So what happens on animal may not necessarily happens in human. So how representative the animal is to human is a question. And also this link to the second point is that lack of mechanistic understanding, because we just observe there's a dose applied, and to observe what's the adverse outcome, we don't really know at that time that why it caused this adverse outcome. And there's also consumer drive, because now more and more consumers are concerning on animal welfare, and also more and more consumers begin to select the vegan and plant-based food. So all of this driving us to change the method of the risk assessment. At the same time, we have opportunities over the last few years. So for example, we have rapid advances in scientific knowledge, such as exposure science or genomics. And also we have huge technology advances, such as high throughput screening methods, informatics methods, computational toxicology method, and so on. And also this speed of innovation, creating novel materials such as nano or biotechnology are also making that the speed of creating novel materials much faster than before. So all of this are basically give us opportunity to change. And so this is what we are talking about, the change. So the change is mainly from the animal-based approach to the next generation risk assessment approach. You can see from left to right, there's a series of publications by different very influential organizations in the world to beginning to talk about this and advise, apply this, and also advocate this over the last many years. And so in general, this NGRA, which is next generation risk assessment, is an exposure-led, hypothesis-driven, and risk assessment approach that integrates new approach methods to assure safety without the use of animal testing. So there are 3 points here. Is that first is that we talk about exposure-led hypothesis driven. So in the animal study, the exposure we applied is the very relatively high dose exposure. And here in NGRA, we used a relatively realistic exposure, and we also calculated not only the external exposure but internal exposure as well, which I will talk about later in more detail. And also we integrated this new approach methodologies, and you do this without the animal testing. So in other words, we use science but not animals to do the risk assessment to ensure the safety of our products. This is the framework that we use or applied in Unilever. As you can see that here we start from exposure, as I mentioned just now. We estimate the exposure and also we collating the existing information. When we estimate exposure, we will consider the user scenario, how the users use these products. And we also investigate the consumer habits and practices in general. And also we find out the applied dose, and we consider the ADME parameters, which ADME is absorption, distribution, metabolism, and exclusion. And so based on this, we generate exposure estimate. And then on the next slide, the next step is here is that we identify the in vitro biological activity. We characterize this biological activity, and we use a number of different platforms in Unilever. And for example, high throughput transcriptomics. We use cell stress panel. We also use tox tracker and so on. And the purpose of this step is to derive a point of departure. Further, if we need further refinement of this point of departure, we can also incorporate the metabolism into this consideration. And then comparing this point of departure with this exposure, which we generated for the first step, we can generate margin of safety, which is the exposure, the point of departure divided by exposure. And then based on this margin of safety, we will find out there's a whether it is risk, whether it's high risk or uncertain risk or low risk or not. So this is the the general framework that we use in Unilever to do this next generation risk assessment. Next slide. So among those, within this framework, we can see that there's a lot of tools that can be applied to estimate the point of departure or to estimate the exposure. So here is the example of the methods that we are using to estimate the exposure, which is the physiological based kinetics modeling. The aim of this modeling is to estimate the chemicals concentration in different organs and tissues in human body after human are exposed to this chemical via different exposure routes, such as oral, skin, or inhalation mainly. And this estimation of the concentration is mainly based on the ADME properties of a certain chemical. As I said just now, ADME is absorption, distribution, metabolism, and exclusion. And in the middle of this slide, as you can see here, there's a schematic of how the PBK model is working. So in the middle here, this block represents a PBK model, which is a series of mathematical equations. And it takes a lot of inputs and it generates an output. So the inputs here, you can see they include physiological parameters, physiochemical parameters, kinetic parameters, product use information. And by physiological parameters, we mean the parameters such as body weight and blood flow, tissue volume, and so on. And by physiological chemical parameters, by physical chemical parameters, we mean that the log P of the chemical fraction unbound in the plasma or tissue plasma partition coefficient and so on. For the kinetic parameters here, we refer to thermal absorption, we refer to hepatic metabolism, we refer to renal excretion, and so on. And for the last one is product use information. This is how the consumers using our products, which will determine the external exposure, that including how much dose has been applied and how frequently it is applied, where it is applied, what's the formulation of this chemical, and so on. And all of those parameters are fed into the PBK model, which will generate a curve of the change of the concentration over time, as represented here. So as you can see here, this curve, just a demonstration, is, for example, it is a change of concentration over time in plasma. And among this curve, we are specifically interested in three, or normally one of the three, or all of these three metric. One is the Cmax, which is the maximum concentration in, for example, here is plasma. It can be in other organs, such as in liver or in kidney or in brain and so on. And here is the maximum concentration along this time course. And then Tmax is time that it takes for the chemical to reach this maximum concentration. And AUC is area under curve, which is the area here. So these are normally three metric that we are interested from a PBK model. And there's a lot of challenges in applying this PBK model, and I just listed two of them. One of them is understanding this ADME mechanism. So how this chemical is absorbed and distributed and metabolized and excreted from within and from the human body. So based on this, because different chemicals may have different mechanism. Some of them, for example, is cleared by liver, some of them is cleared by kidney, some of them have some transporter effects, and so on. So we cannot use a same model to apply at all the chemicals. So we need to investigate for each specific chemical what is specific mechanism. And based on this, we need to tackle another challenge, which is the parameterization of the model. So for example, here we have so many parameters, and by parameterization means that we need to provide a value for each and every of those parameters. How we do that? We cannot focus on all the parameters. So many of those parameters are generated by in silico predictions, but also and also this is our tier one, or the beginning tier of the PBK modeling. And then if we want to refine it, we're going to either generate some in vitro data, or we're going to select some in vitro data from the literature for some important parameters. The importance here means that the change of this parameter will have a big change on the point on the exposure in the end, on the Cmax or Tmax or AUC. And then this is the second tier, which is in vitro parameterization. And for third tier, if it is still not satisfactory, then we will also, for example, use some clinical study data to see whether this prediction is as expected or is close to the clinical study result. For the clinical study, so by comparing this, if it is not close, then we'll go back to see that for those impactful parameters, whether some of them or one of them or some of them needs to be, the value needs to be updated. Then in this case, we need to again look at its ADME mechanism to see that to what extent this needs to be changed and so on. And in the end, we'll get a result which matches the clinical studies. So this process is quite it's a quite challenging process, because we not only consider the for example, the clinical study, we also need to consider the mechanism of the chemical, because mathematically you can change the value of many parameters, and it will generate a very good fit of the clinical study. But sometimes or many times that is not reflecting its ADME mechanism. So in this case, we need to change or adjust the parameter, the value of the parameters according to this ADME mechanism. So this is about the PBK modeling. And another part, which is dose response modeling, is the modeling that need to be that will be used to generate the the point of departure. So the aim of this is using the dose and response data from a certain in vitro assay to derive a point of departure regarding a certain biomarker after exposure to a certain chemical. And also because we're not doing this for a in vitro assay, we're doing it for a number of in vitro assays. So we need to combining this point of departures from different in vitro assays regarding different biomarkers. And the overall bioactivity of the chemical can be described by looking at this combination of this point departure from different sources. And then this point of departure can be then compared with exposure from PK modeling, as mentioned just now, and so that a safe decision can be made. And in the middle of this, you can see there are three very typical dose response curves. For the first one, you can see that here there is a clear dose response. So for example, this is the concentration of a certain chemical with its increase. This how to say vertical axis is the fold change of a certain biomarker. So as we can see here, when the concentration is below 0.1 micromolar, there's not much happening in terms of the fold change. But when it is above 0.1, you can see there's a significant fold change happening. And this one is different from this one, is that with the increase of the concentration, as you can see here, that the change is still, for example, here, the fold change is still below 1.2 or around 1.2 fold. And it is quite difficult to say whether it is really a fold change or it is just a noise in the assay. So in this case, we cannot say that there's dose response really. And then in here, this is another extreme case, which is that we cannot see any dose response. You can see here, even when the dose is increases to 1,000 micromolar, there's still nothing happened. So these are the three key, typical dose response curves. And from these three different dose response curves, we can see that there's a lot of challenges. The first one is that whether there is a response. So for example, here and here, it's quite certain. But what about here? So we cannot say there is or there is not a dose response. The second challenge is that at what dose there's a response. So we need to find out the point of, basically the point of departure. And the third point is uncertainty, because as you can see here, this is not a, we cannot say that this happens at a single dose. So we need to characterize its uncertainty as well. So this uncertainty can be, again, characterized in many different ways using many different methods, such as Bayesian methods and Monte Carlo simulation and so on. So these are also a challenge to characterize the uncertainty of this point of departure. After we generate this point of departure for a certain assay regarding a certain biomarker, as I said just now, then we can see that these are the example of the point of departure from different assays across different biomarker. And we just plotted them together with uncertainty characterized. And then this yellow band is the exposure. Then we compare this point departure with the exposure, and then we can get the margin of safety, or we can also call it bioactivity exposure ratio. So based on this, we can see whether for this chemical, this certain exposure scenario, whether it is safe or not. So the previous slide is about the methods and NGI framework and the methods that applied in the NGI framework. And from this slide, the next one, I will introduce very top level, introduce a case study which demonstrate how to use those methods in the NGI framework. So this case study is about sulforaphane, and which is a naturally occurring compound in some vegetables, some cruciferous vegetables like broccoli, like cabbage, and so on. And in the food, it is an inactive form of glucoraphanin. But when the vegetables are destroyed, basically, for example, chopped or chewed, there's an enzyme will released, which is called myrosinase. This enzyme will release, and this will come into contact with the inactive form of sulforaphane, which is glucoraphanin, and then in the end, the sulforaphane will be formed. This is how sulforaphane is formed. And then sulforaphane has been associated with various health benefits and may beneficially affect the cancer, heart disease, diabetes, and digestion. So that's why we use this hypothetical case study to show that if there's a food supplement with a dose of 60 milligrams of sulforaphane, then whether it is safe for people to take. So this is the a very top level case study. We start from exposure. We assume that a supplement that contains 60 milligrams of sulforaphane per tablet is taken once per day. And then we build a PBK model to estimate this corresponding plasma concentration. And as I said, parameterization is a big challenge of PBK model. So most of these parameters needed for PBK model are based on in silico predictions. And we do use the in vitro assays to find out the fraction unbound and also the apparent permeability coefficient, which is PEP. And then we base on all of those parameters, we run this simulation over 7 days. As you can see here, this is roughly how the plasma concentration looks like. So as you can see that for each day of application, this concentration is quickly in the plasma, is quickly increases, and then is quickly cleared. So there's no obvious accumulation in the human body before the next dose. So after this, there's 7 doses here. And the Cmax, which is here, which is the concentration here, is 0.84 micromolars. This is how we calculate the exposure, and how we calculate hazard is that we estimating the variance of point departure across based on the in vitro bioactivity data using 3 of the in vitro bioactivity platforms across the platforms and across biomarkers, such as high throughput transcriptomics and cell stress panel, and in vitro pharmacological profiling. So, and from this, we can see that we can get different point of departure of sulforaphane. So some of them is from high throughput transcriptomics. Some of them, I'm not sure you can see that this is from cell stress panel, and some of them is from in vitro pharmacological profiling. So here, for simplicity, we just take it the lowest point departure as 0.072 micromolar. So this is also a very conservative estimation of point of departure. So as you can see, the Cmax is about 10-fold higher than the lowest point of departure. So in this case, we can see that the proposed dose could not be supported. But we cannot stop here. We're going also need to refine it to see whether there's some area that we can further investigate into this process. Is that we refine the clearance? We can refine the clearance in the PBK model, so that here, most of the parameters, including the clearance, is based on the in vitro in silico predictions, and we know that clearance is quite influential parameter because it has a big impact on the Cmax. So we can refine the clearance in the PBK model. What we can do in the point of departure side is that we can also identify the relevant pathways to investigate further to see, or for example, we can also for example increase the time points of the measurement, increasing the different doses, and so on, to find out whether this response, this point of departure, is adaptive response or it is adverse response. We also can incorporate in the uncertainty analysis, and whether, because as here we took the the smallest point departure, we take the point estimate of Cmax, which is not ideal. So we can go further to estimate this as well to see whether which whether they have any, what they will have uncertainties, whether this uncertainty will influence our decision as well. So these are quite top-level introduction of this case study, and the aim of that is to show that roughly how the methods can be applied. Next slide is about a summary. So as you can see here, that the hypothesis of this NGRA is that if there's no bioactivity is observed at consumer relevant concentrations, there can be no adverse health effects. And this is what we're talking about just now in terms of whether it is adverse or adaptive response. So at the moment we say that whether we base our risk assessment on any bioactivity bioactivity. If we need to refine it, we go further to find out whether it is adverse or whether it is adaptive or adverse. And then the second point is that at no point does NGRA attempt to predict the result of high-dose toxicity studies in the animals. So basically, we only use the consumer-relevant concentrations. And the last point is that the NGRA used new exposure science and understanding of human biology, which is based on mechanism understanding. So the way forward, there's a different ways forward, of course. Science and technology, there's a lot of area we can develop. For example, in vitro assays, such as cell viability, such as genotoxicity, and so on. In silico tools, such as QSAR models, read-across, which already demonstrate in the previous presentation. And also the ADME, study of the ADME of the parameters and so on. And also we can talk about the organs on chips, which is already under a lot of development nowadays. We also can make efforts on the weight of evidence approach, and we to combine different lines of evidence according to their weight. And we also need to find out the uncertainty analysis is also quite important. Communication and improving acceptance is another area that needs a lot of efforts in addition to the what we call the hard part, which is science and technology. This is soft part. So to ensure this, to facilitate this communication and improving the acceptance, we can improve the reproducible and the transparent in the in vitro assays and in data analysis. We need to clear document the applicability domain and uncertainty and limitations. We need to involve different stakeholders to sharing and evaluating. This is quite important of the safety and decision maker using NAM approaches in food ingredients risk assessment, identifying their strength and weakness. So basically, not only Unilever or not only, for example, EFSA or FDA, we need to come together to evaluate those methods and to see whether where its strength is and where its weakness is and where is their applicability domain is. And we're developing networks of organization with common interests. We're advocating, educating, upscaling for different areas for different audience. The last bit is that this is what we're doing in China.
他们说不说了，到点了啊，敲了3分钟了啊。好。
Okay. So So thanks and yeah, thank you.
好，感谢汤博士啊，因为已经到点了，这样吧，我们还是如果有问题的话，我们问一个问题，好吧？现场的话，我们是会由彭老师来主持。有问题吧，大家？线上的可以举手，或者是聊天室啊。我刚才好像听到问题了。
请问哪位有问题？好，贾教授，我们会长这边没有什么问题，好吧？
好啊，那这样没有问题的话，我们就因为时间的关系呢，我们就下一个报告了。好，谢谢汤博士啊。
好，
哎，
我看这个怎么停下来啊？等一下。
好，你把退出共享啊。好，在退出共享的时候，我是介绍一下下一个报告啊。下一个演讲人是 Gilbert Schonfeld 博士。Schonfeld 博士呢，来自德国联邦风险评估研究所，也就是我们大家都知道的 BFR。他是实验毒理部的主任，同时呢，也是德国柏林查瑞特大学医学院实验毒理学和替代方法领域的教授。此外呢，他还是德国实验动物保护中心的负责人。Schonfeld 教授的研究的重点呢是替代动物实验的方法，他还致力于毒理学评估策略的开发。他今天报告的题目呢是用于消费者健康保护的非动物性测试方法的开发现状及科学挑战。因为疫情的原因呢，Schonfeld 博士也是通过线上 和大家交流。下面呢，我们就有请上Feld博士。Hi，上Feld博士 ，please。
Thank you very much for the kind introduction. Just a short feedback. The translation has been switched. That it's in Chinese when it should be in English. So nevertheless, I hope you can hear me and you can see the presentation. So are you able to follow the presentation?
Yes. Okay.
Perfect.
It's okay. Yeah.
Good. So good morning to those who are in Europe. Good afternoon to those who are in North America or in China, or good evening. My name is Gilbert Schönfelder, and I'm presenting the German Federal Institute for Risk Assessment, which is located in Berlin, the capital of Germany. I'm totally appreciate that I could be with you at the Chinese Food Security Conference 2022. And I'm also the head of the German Center for the Protection of Laboratory Animals, which is a government-run institution. The title of my work is Current Developments of Animal-Free Test Methods for Consumer Health Care. Current developments of animal-free test methods for consumer health protection and their scientific challenges. And I'll give you a short introduction about us. Who we are. First of all, the German Center for the Protection of Laboratory Animals was founded in 2016, according to a call by the German Minister for Food and Agriculture. And what we are doing is we are located at the German Federal Institute for Risk Assessment. And here we coordinate all activities, not only within Germany, throughout Germany, aiming to, first of all, reducing animal experiments to the necessary minimum and provide the best possible protection for laboratory animals. And what else are we doing? The work of the BFR intends to stimulate national and international research activities and promote the scientific dialogue regarding the implementation of the so-called 3R principle by Birch and Russell, which is the meaning of, first of all, replacement of animal testings, refinement, and reduction. So, but you might be more interested why I'm talking on this conference. And again, the Federal Institute for Risk Assessment is the leading institution in Germany and one of the leading institutions in Europe, which addresses consumer healthcare protection. So we're addressing many topics where, of course, testings are being involved, which are related to toxicology. And therefore, I just want to give you a short introduction in the German Center for the Protection of Laboratory Animals. We have 5 areas of competence. I don't want to go through all of those, but just to give you an in short overview, the main one which I present today and I like to talk about is the competence area for 3, which is alternative methods and toxicology. And here we develop novel test methods, and we are part of an international coordination. Why are we a part of international coordination? Because as we are a governmental institution, we have to fulfill laws. That means fulfill the European directive, but also the German directives and laws, according not only to the German Animal Welfare Act, but also to the protection of consumer health care, which is related to laws related to the regulation of pesticides, chemicals, and of course to biocides as well. And we wouldn't be sitting here together if we wouldn't test those chemicals to address whether they can lead to severe health effects. And to test those, what we need is a biological experiment. And in general, this has been performed in vivo by using animals as a model which could mimic human disease, or where we could find out whether the exposure, for example, of chemicals could lead to adverse effects in humans. So therefore, there is a coordination worldwide, and this is addressed to the OECD. And the OECD coordinates the national network for the assessment of regulatory significance and suitability of all alternative methods proposals for the validation in line with paragraph 16 of the German Animal Welfare Act. This is part of us where we are located into the OECD. And we here are part of the test guideline program for animal-free test guidelines. So the section or competence area alternative methods in toxicology has been built up by an expert group of external reviewers, which could advise us in the work we are fulfilling. We develop test and assessment strategies for the assessment of chemicals. And of course, we do our own research and development, and therefore we develop our own and for the market, the novel in vitro test methods. To give you a short introduction where we are located in here.
Can you show
So I
your
was talking
slides on
about the
full screen?
OCT test guideline program on the worldwide level. However, there is a similar system regarding in Europe, which is a Pareo network, which advises the regulatory relevance and suitability of new test methods and the selection of new methods for the validation that they can go into the regulation assessment of, for example, chemicals. So chemicals, for example, which would be marketed or imported to the European market and can lead to an exposure to European citizens. So therefore, we here at the BFR or the BF3R, which is the abbreviation for the German Center for the Protection of Laboratory Animals, here we have located the national coordinator in the competence area of alternatives to test methods to toxicology. And so far, this person is in his personal role has been involved in the OECD test guideline program for the development of animal-free test methods in toxicology. So what we are doing again, we do development of assessment strategies. We have in-house expert groups, and we have our own test methods development, which we try to get into the OECD test guideline program or Pareo network as well. So Germany is part of the OECD, and therefore we're taking part in the development of test guidelines for the purpose for consumer healthcare protection. So what is Pareo? You might be not informed what is on the European level besides the OECD level. Pareo is the Europe ECMA network for preliminary assessment of regulatory relevance. ECMA is the European, 3R Center of the European Commission. And here, new methods pending validation, which have been submitted to ECMA for assessment or validation, are checked by the competent authorities represented of the European member states for their proper relevance. And as we are a member state of the European Commission, European Union, so we are a part of the Pareo network. So the process is called Pareo, which is again preliminary assessment of regulatory relevance. The Pareo network is a network of national regulators that provides ECMA with upstream input and preliminary views on potentially regulatory relevance of methods or approaches submitted to ECMA for the validation and or for peer review. So at the European level, the process of the acceptance of methods is conducted in similar manner to that of the OECD, but in most cases, OECD test guidelines are adopted directly as part of the regulatory adjustment of the European Test Method Regulation 440/2008. So this is Pareo, and as I said, we are part of the safety assessment system at the OECD level. So OECD again, but you're all familiar with it, is the Organization of Economic Cooperation and Development. So it addresses environment, health, and safety program. And of course, there's one section, which is the part which I would like to talk about, is the chemical safety and biosafety. So part of the OT test guideline program is, of course, the representative of each of the member states of the OECD, and this is given by the national coordinator of the OECD test guideline program. And the German coordinator here is Michael Oergelschläger, which is a colleague of mine, and is working at the German Center for the Protection of Laboratory Animals. So he's dealing, and we are dealing with the OECD test guideline program and with the OECD principles of GLP and compliance monitoring procedures. And this brings together the mutual acceptance of data. So again,
Dr. Schamfelder, Could you
OECD
hear
is
me?
addressing also environment, health, and safety, but the test guideline program, which we are part of, section 4, which means health effects. So we are addressing human health effects and not the environmental fate in our work. So one of the parts which we are doing is that, of course, we are going and here we each of well, every scientist and companies can submit their proposals for new test methods. So we are driving them through the validation step, to the test guideline development step, and to the regulatory acceptance. So those who are interested can submit their proposals, which are called SPSFs. In the next one of the next slides, I will present them to you, to the national coordinator, and he suggests whether they can get into the O test guideline program. So how does it look like? Just to give you a short introduction. Of course, you're a test method developer. You want to get this test method been validated and accepted for the OT test guidelines. So it's been submitted as an SPSF to the national coordinator, and he is submitting it from the national level to the international level to the OECD secretary. And here it comes into the compilation of comments. So there is a commenting procedure between the working groups of the national coordinators. And from there, the inclusion into the work plan is then it comes to the OECD expert working groups. And from there, they are having a lot of discussions to the drafting and the revision of the test guideline, which is proposed to become an OECD test guideline. And the compilation takes always a little bit while. And then after the commenting and the drafting, then it goes back to the working group and to the national coordinators, and from there to the CBC, which is the ministerial representatives. And from there, it gets its TIGI adoption. And if successfully everything worked well, then it's been accepted at the end as an OECD test guideline. So you might have heard already today about strategies, which are more or less new test strategies. And one of the test strategies I want to introduce to you is, of course, the adverse outcome pathway. That means that's the development of assessment strategies. And the adverse outcome pathway concept, AOP, is according to the OECD guideline 184. And what is the main purpose? It has been to combine, to replace animal experiments to regulatory toxicity testing and assessment. And therefore, it's been separated in different steps, and all of those focus on the mechanisms of adversity. And the chemical structure is the first focus, and then we have the molecular initiating event, for example, receptor binding, DNA binding, or protein modification. Then we have the next step, which is a biological step further, which is on the cellular level, that there are mechanisms being addressed like gene expression, protein expression, or signaling pathways. And then it goes to the next level in the biology, which is on the organ and tissue level response. So your endpoints have been addressed to the physiology, to homeostasis, development, and to the function. And at the end, it rests, of course, the main task, which is the adversity. So it has to address whether an organism population has a response, which is leading to an adversity. So it means to a disease like allergy, malformation, cancer, death, reproduction defects, developmental defects. And all of those is again been addressed at the collaborative adverse outcome pathway wiki, where you can take a look of it in there and can see which AOPs are already been highlighted and what kind of key events are already addressed by separate test methods, which is mainly on the in vitro level. So again, here showing you the summarized is then that through all the different levels in the biology leading to the end of the disease, that means the adverse outcomes, you have separate key events, which you separately analyze and by separate test methods. And then like a sum up of all those results, you can conclude from there if this leads to an adverse outcome or not. And for those who might not be familiar, then you might take a look at the AOP wiki, which is public available. And it's called Welcome to the Collaborative Adverse Outcome Pathway Wiki. You see there different subsites of the website, which is, of course, the view of the content, which means they show, you can look for AOPs, for key events, for key event relationships, and for prototypical stresses. But you can also contribute to new AOPs. So you can start to put a new AOP in there. You can participate on the development of a handbook, but you can also get different information out of it. And you can be involved with the community who's developing AOPs, and also different test methods to fulfill one of those AOP strategies by being part at the AOP forum, third-party tools, or AOP help desk, for example, where you can interact. So you might like to know what we are doing, because we are following this kind of idea of the AOP strategy. So since 2016, we're the governmental institution, the German Center for Protection of Laboratory Animals, was founded as part of the BFR. We mainly address the part of replacement strategies. So you have heard in the former presentations about the NAM projects. So all of us, what we are doing is standardization, validation, and development of test strategies, and therefore we build it up in bioimaging and screening center. So we are able to do high resolution imaging, of course, liquid handling, plate handling, and high content imaging, which brings together that we are in a high throughput chemical analysis. That means we build it up also a chemical library. So currently we are investigating 440 industrial chemicals, biocides, and pesticides. And the endpoints we are currently addressing on the estrogen, androgen, and thyroid-mediated activity. That means we are focusing on endocrine disruptors. And as you can see, this is part of all which is related to, for example, the European Endocrine Disruptor Program, of course, to the OECD thyroid hormone signaling program, to the US NICOTAM and reference list of endocrine disruptors, but also of the US ToxCast endocrine profiling and EFSA pesticides program. So those chemicals were selected, 440 industrial chemicals we are currently analyzing on test strategies and methods which we are developing. And you might be interested what we are currently doing, or what are the endpoints we are focusing on. And this is I represent my colleagues which are working on it. So we have one of those projects of the BFR is called Cancer ODC project. And this means that we try to identify endocrine active substances leading to chromosomal instability in cancer, which means one of those endpoints which we newly bring in is genotoxicity related to endocrine active substances. And here we have the molecular initiating event, which is, of course, a coupling to an estrogen receptor. And then we separated the adverse effect, which is at the end cancer, into separate key events. So we are looking for centrosome amplification, pseudo metaphase, lagging of chromosomes, and whole chromosomal instability. And we're studying those effects in colon epithelial and colorectal cancer cell lines. Here we try to develop a test strategy which might in the future probably get into the OECD, but we're at the first step that we try to understand the system. If you are interested in it, I can just highlight it. You can go to the BFR homepage on the English side. And here we have a scientific-based journal, which is called BFR2Go. And there is an article highlighted which addresses this topic about chromosome instability and cancer, and the identification of those instabilities and cancer by substances which have endocrine activity. So this is one of our projects, which is called, again, Cancer EDC project. Coming to the next project, this is another one. This is also highlighted in the BFR2Go. scientific journal. I mean, we published all of those already internationally on, for example, Environmental International, but also in iScience and current protocols and things like that. So you can go into PubMed and get those information out, but also read this kind of short introduction for the for the public, which is the BFR2Go magazine. And what we're doing here is that we try to identify substances, whether they have estrogen activity, that means in total endocrine active substance, out of this library which we are analyzing. And here the endpoint is simulation of cell adhesion and cancer. And we were able to identify an effect on breast cancer cell lines, which was leading that we have revealing a bubbled structure in the cell membrane, which you can see here on the red side. It means if we have treated breast cancer cell lines with anti-estrogenic conditions, for example, with a substance which has been used for treating patients, the most common one is fulvestrant or Faslodex, which is the commercial name. Then it leads to an anti-estrogenic effect, and this anti-estrogenic effect is leading to an activity of the estrogen receptor alpha, or blocking it. But this leads to a dislocation of adhesion molecules. One of the prominent adhesion molecules is ecatirin, which was tagged into with GFP and overexpressed in cell lines. And as you can see on the right-hand side, you see those green dots within the cell membrane, and that is in the cartoon downstairs the accumulation of cadherin protein junctions between the two different cells. And when you on the other side antagonize the anti-estrogenic effect with estrogens like simply estrogenic compounds like estrogen, or even other compounds like DES, or chemicals which are known to have estrogenic effect, then there is the way that these bubble structures are getting lost, and that means the stiffness of the cells is getting lost. And if the stiffness between two cells getting lost, they can lead either more to metastasia. So the effect which we are analyzing is actually a phenotypic readout, but this has not only the purpose that we can analyze chemical compounds, also we have an endpoint which is probably mostly relevant for humans, because we found the same kind of endpoints in tissues of women with breast cancer. And we know from atomic force microscopy that cells which have a bubble structure, an anti-estrogenic effect, are more stiffness, so they cannot leave a cell which is adverse cannot leave the cell-cell interaction. However, when the bubble structure is getting lost, and that means we have not a deeply organization of ecatirin anymore, then it's been able for the cell to leave the cell conjunction, and that's for how the metastasia tries to develop. So this is in the context of EMT, which is epithelium mesenchymal transition. So again, with this endpoint, we have a high throughput screening developed, which is called EMORPH assay, and hereby we can by in phenotypic readout really screen compounds for estrogenic and anti-estrogenic effects. So one of those projects, which is a development now of the NAM project E4 assay, is of course that we want to get more insight. So the EMORPH assay was the first step into, and now we try to standardize this kind of technology, and therefore we need to get into a validation step. So this is a validation step which we submitted to Pepper. Pepper is a public-private platform for the pre-validation of endocrine disruptors characterization methods, which was founded in France, and this is an institution which is located in France, which has the purpose to develop new tests for screening of endocrine disruptors. And here we submitted it in 2023. We probably get into the first pre-validation step of our development of the phenotypic screening and the prediction models and similarity research. So this is a combination of work which we are doing with local partners. One is the University of Saarland, which is one of the federal states here in Germany, but also with the FMP, which is a research center located in Germany, especially in Berlin, in which addresses pharmacology. And therefore we want to get a more sophisticated and precise screening system out, which is able to screen chemicals for the purpose of estrogenic activity or anti-estrogenic activity. So this kind of test can also be used for pharmaceutical companies as an animal-free test method. Okay, coming to the last one. I don't want to go into much details here. As you can see, this is a project which is addressed to the thyroid hormone signaling. So it's all about the identification of substances with activities on the thyroid hormone system. So here we address several steps in the biology of the thyroid hormone system to get an AOP at the end out, which is fulfilled with separate test methods. So one of those is, of course, the dehalogenase activity. The other one is the deodenase type 1 to 3 activity. And the other one is the monocarboxylate transporter 8 activity, which all have to be a part of the whole system that you understand at the end how thyroid hormone adversity can be developed. So one of those besides is that the thyroid hormone synthesis has also been analyzed, and here we analyze the tyroperoxidase activity and the sodium iodide supporter activity. So at the end, we hope that we have for an AOP to address the thyroid hormone system at the end, different test methods been developed, which address separately different key events, which are important to understand at the end, the outcome, if we screen substances. And this all has been funded, of course, and of fundings from third parties, which is, of course, the Panoramics funding, the Athena funding, and scenarios. And the validation has been submitted already to NETVAL, which is called European Network Authority and Validation Study. And the proposed is also with PEPPER. So we try to get into PEPPER the second kind of pre-validation in there. Besides EMOF assay, now we have a thyroid, part of the thyroid project being submitted to PEPPER for pre-validation that is getting to the OECD. So I'm coming to the end and try to just highlight a couple of more. One is that besides the non-genotoxic carcinogens, which we addressed already, that we have another project running, which is Risk Hunter 3 project, and this is a cell painting. Cell painting means that we try to understand how different chemicals can change the morphology of cells, and at the end, which leads to a phenotypic change, and this kind of phenotypic change stands at the end for an adversity which might be scaled to a human disease. So Risk Hunter addresses a 4-eye staining system, which is an iterative indirect immunofluorescence imaging system, which is then run by our facility, which is able to do high throughput screenings. So coming to the end now, the last slide is that one of the parts which I like to present to you is physiology, which is mainly not addressed currently in toxicology, but is highly relevant and which is published in 2022 and 2020 already. So we address the circadian rhythm, and what we found out is that restoring the circadian synchronicity can improve the physiological relevance of in vitro test methods. And as you can see here, in general, you have the in vivo situation, and there is a circadian rhythm which is responsible for our physiology, as you know, and when you leave the country and go overseas, you have a change in circadian rhythm, and this can affect your physiology. But what is not so much known is that when you do in vitro analysis, that each of the cells within one cell plate has separate circadian rhythms. And this leads at the end to a point where the dose response curve of each of the cells is differently, but in totally you try to sum up an average response of all the different cells, which is, all the same cells, but with a different circadian rhythm in one of the cell plates. However, we ask ourselves what happens if you synchronize all the cells that they are within the same circadian rhythm, like in the in vivo situation. And in this case, we were able to show that when you synchronize the cells, so that when you bring all the cells in one cell plate of the same cell type, the same circadian rhythm, then you have a tremendous increase of the cellular response of compounds. So for example, TCDD, which is of course a typical compound dioxin, which does an induction of cytochrome P451A1 induction, you can see that with synchronization, we have a tenfold increase in the cellular response. So what we can, to sum up here, what we can show is that each of you who is addressing in vitro test systems should ask whether it's bringing in the circadian synchronicity would be able that you at the end are more precise and you have higher responses. You test compounds and you see the dose response curve. So I'm coming to the end. I would like to thank you for your attention. And if there's a short time for questions available for you, then I'm happy to take those questions. Thank you very much for attention. And thanks again for the organizers for inviting us to present the German Federal Institute for Risk Assessment. If you want to follow us, just go on Twitter. BF3R at Center is one of our Twitter site where you can follow what we are doing currently on the day-to-day basis. Thank you very much.
Okay, thank you, Dr. Schoenfelder. By the way, we do have a simultaneous translation. Maybe you can try again. Okay. So 大家有问题吗？看现场有没有问题的吗？刚才因为我跟他交流的时候啊，他说他好像没听到翻译啊。大家如果有问题的话，请用英文啊，尽可能用英文。现场有没有提问题的？线上的老师
有问题吗？各位
Perfect. Thank you.
线上老师
贾教授目前没有反馈。
好，谢谢。
好谢谢
线上老师有问题吗？好 ，No questions. Oh, thank you again, Dr. Schoenfelder.
For sure.
OK， 好，这样的话，我们就是到了茶歇啊，茶歇。当然，这样请大家3点50回来。好，谢谢。
谢谢，你好。
谢谢 ，Thank you. Bye.
我们下面我介绍下一个演讲者，他是欧洲食品安全局局长办公室分子毒理学首席专家 Dr. George Kass。他今天报告的题目是欧洲食品安全局关于非动物方法的路线图和相关案例介绍。下面我简要的介绍一下 Dr. George Kass 的基本情况。Dr. George Kass 他是在1990年在斯德哥尔摩大学斯德哥尔摩Karolinska 研究所获得的这个博士学位，后来在瑞士联邦技术研究所进行博士后研究，随后加入沙里大学，成为沙里大学的全职的毒理学的教授。Dr. George Kass 他曾先后发表100多篇在毒理学领域里的论文，他目前是毒理学和应用药理学杂志的副主编。下面有请 Dr. George Kass 给我们做报告，有请 ，please.
Right, thank you very much, and good afternoon, everyone, or good morning for the people who join from Europe or the US. So first of all, I would like to say thank you to the organizers for having invited me. We're very grateful about that, and for giving us the opportunity to share with you some of the work we are currently doing in the area of NAMs. You already heard from the previous speakers about the initiatives that are ongoing in Europe, and I will carry on in that vein. But taking the approach that we as risk assessors are taking, and the challenges and the opportunities that come to us in the form of NAMs. So I will start sharing my screen, and with the, okay, it's working. Right, let's hopefully, yes, I can see, yes. It's also showing the right format. So the title of my presentation is EFSA's roadmap on NAMs, and presenting some case studies that are linked to the use of NAMs. And I'll explain a bit more about why we're investing very much into case studies. See if I can go to the next slide. It's working, yes. So I also have to share with you a disclaimer. So what I'm presenting here are not necessarily the views, thoughts, and opinions of my organization. And just to give you a brief overview about where I'm coming from, and a bit about the organization that is EFSA. So EFSA is one of the agencies that working together with the Commission, the European Commission, and where the role is really to look at the safety of the consumer. So EFSA does it for all the chemicals and also biological agents, microbiological agents that are involved in food safety, and as well as feed safety. Whereas, for example, our sister agency, EMA, is responsible for all what concerns drugs and veterinary products. ECHA, on the other hand, up north in Finland, in Helsinki, deals with industrial chemicals, so both on the production in Europe as well as the import. And ECDC deals with diseases. We are located in the south of Europe, so we are located in Italy, in the first third of northern part of the country. This slide, which we call also the EFSA Atom, gives you an overview really of our tasks and what we are active in, our remit. And you'll see that starting from the top left there, that we're really dealing with what we call from farm to fork. So we're dealing with plant health. So this is to address pathogens, to address pests. that affect the plants and the health. We're dealing with plant protection products also involved to maintain plant health, but also can be used as herbicides to get rid of certain plants. We deal with GMOs. We deal with food that is used for animals, so animal feed. Our role is also the health and the well-being of animals. We also are involved in looking at hazards, microbiological hazards that could have an effect on human health, so it's the so-called biological hazards. We're dealing with chemical that we find in the food, but not intentionally added to food, so we call them chemical contaminants. Whereas we're also dealing with chemicals that are added intentionally to our foods. One example here is the food additives, for example. And also we're taking, within our remit, we deal with materials that are used to protect the food, to be wrapped around food, so the so-called food packaging materials. And we're also dealing with nutrition, so we're addressing issues such as values for vitamins, for oligominerals, but also the consumption of sugars, the consumption of salt, etc., just to come up with some guidance values there for human nutrition. One of the common aspects you will see here is that a lot of those sectors involve chemicals. So chemicals are at the core of those sectors, and it's a chemical safety that is the role that EFSA is playing here. So EFSA is the European organization that deals with that provides a risk assessment on chemicals. And we are not doing the risk management, so not the risk managerial task is not included in EFSA's remit, but we also are very much active in communication, so the communication and the collaboration with other organizations. When we look at the chemicals that we're dealing with in all the different sectors, we can divide them into two. So the so-called regulated products, so these are chemicals that are meant to be put into the food intentionally. And typically behind them, you have an applicant. There's a company, there's industry that sends us a dossier where they provide us with all the information that is needed for the risk assessment of those regulated products. As mentioned already, we're also dealing with contaminants in the food and the feed chain. And here, typically, there is no industry, there's no applicant behind it. And because of this, the type of information we get is quite different. So it's not from a dossier, but it's information that we either get from legacy data on compounds that was previously accepted on the European market, or we're taking the information from the public literature, from published papers. And about the chemicals that fall under the regulated products, of course, some are new chemicals, but we're also dealing at the European level with common chemicals that are already on the market and for which there is a legal obligation to do a reevaluation and a reassessment of the safety of those chemicals. Typically, this would apply for plant protection products where have a 10-year cycle or 5-year cycle, depending on the plant protection product. What is also typical on the EU scene in terms of the data that are required to be submitted for the regulated product is that in some instances, such as, for example, the plant protection product, the legislation, there's a regulation that stipulates the data to be submitted to EFSA, whereas in other situations, we deal with guidance documents that inform the applicants about what type of data should be submitted. I'll spend a bit of time on this just to explain the situation in the EU, and I'll start with the plant protection products. So here's the Commission Regulation 283 of 2013. And what you'll find, the information you find in that regulation is that the information to be submitted should be sufficient to evaluate the foreseeable risks to humans, animals, and the environment. And moreover, the legislation in one of its annexes provides you with the list of studies that have to be submitted. These are your typical toxicological and metabolism studies, and you will recognize your traditional toxicity studies, mainly done mainly coming from in vivo studies. The situation is quite different when it comes to other sectors, for example, the food additive sector. Here, the legislation only stipulates that food additives must be safe when used. And for this, EFSA has then the possibility of developing specific guidance, sectoral guidance documents, informing the applicants how to do the evaluation, or how the evaluation of food additives is being done, and what data are needed to do this evaluation. Coming back to the data that we receive at EFSA's level, and here I'm primarily focusing on the regulated products. And you will see that they would consist of biological studies, primarily focusing on the ADME studies, so looking at the behavior of substances in the body, metabolism, absorption, excretion, etc. The toxicological studies, ranging from generally subchronic to chronic, and also reproductive and environmental studies. And what you'll see there, both set of studies are primarily from in vivo studies. A set of data come from in vivo studies, and they have to follow OECD test guidelines and GLP criteria. The only in vitro studies that we tend to get are on the genotoxicity. And while they're also following the OECD test guidelines, our recommendations are that if the in vitro studies are sufficient to give a clear answer about the genotoxic potential of a compound, then they do not need to be followed up by in vivo studies. So the take-home message, what I want to share with you here, is that the traditional chemical risk assessment relies mainly on animal bioassays. So what is EFSA's approach to NAMs and the use of alternative approaches? So I'll start with giving an overview of what has been happening over the past 2 decades, and where the in vitro test approaches have already been firmly engraved in EFSA's recommendation is in the genotoxicity testing, where an established battery of in vitro tests exists and is recommended. And if there is no evidence of genotoxicity, no further in vivo studies need to be carried out. We also have the TTC approach, which strictly speaking is not a NAM as such, but it relies on legacy data produced over the past 20, 30 years, which allow us to predict exposure levels to chemicals when we do not have the toxicity data to allow us to predict the toxicity. Or allow us to use thresholds where no toxicity is unlikely to, where toxicity is unlikely to occur. This is mainly done for impurities, metabolites, degradation products, pharmacologically active substance present in food of animal origin, and also it's the approach that can be used for exposure to multiple chemicals. And in 2019, we produced a guidance on the use of the TTC. We also use the read-across, which is a very powerful approach that is currently seeing a lot of developments to be used in regulatory risk assessment. It has been used by EFSA for flavorings, and we also use the read-across for combined exposure to multiple chemicals, and on an ad hoc basis, it has been used for food contact materials. One example I want to share with you, again in the area of plant protection products, I mentioned that the legislation are requesting a whole set of in vivo data to be able to do the risk assessment, but we must not forget that each active ingredient, each active substance can lead to the production through its cycle to from application to ending up with the consumer, can end up with a number of metabolites that are not characterized from a toxicological point of view. They can simply emerge from the production by soil or by the plant itself, so they cannot be tested at this stage. And for this approach, we used for the genotoxicity also alternative approaches, so read across as well as the TTC for this compound using the threshold for genotoxic compounds. So this is an example where we use the combination of read across and the TTC. So what is the future? What are our new projects? What are the new challenges? And what are our new ambitions? And here we'd like to start with sharing with you the position in Europe, which has evolved considerably over the past, it's going to be soon 5 years, with the Green Deal, which was announced, and more recently in 2020, the so-called chemicals strategy for sustainability towards a toxic-free environment. And if you consult that document, you'll find that there's emphasis on moving away from the dependency on animal testing and to improve the quality efficiency for the chemical and chemical hazard and the risk assessments. And really it's clearly stated that the Commission will try to move away from animal testing. This is also reflected in EFSA's own strategy, which was also published recently. That's we're talking about last year. So the EFSA strategy 2027. And again, in one of its key objectives, it is clearly stated there, you find it stated that EFSA will develop and integrate new scientific development focusing on NAM-based methods and the minimization of animal testing. This is also to bring into effect the already existing EU legislation on the 3 R's, trying to minimize the use of animals. And one of the key actions is really to develop and integrate new approach methodologies and omics for regulatory risk assessment. So what are we currently doing on that front? I mentioned already the use of read across. Now we're investing currently considerably into the read across through the development of a guidance document on how to use read across in food safety assessment. So this work is under development, it's ongoing. And one of the areas we currently investigating for development of this guidance is first of all to see how we can use read across from a regulatory point of view for chemicals that are within the remit of EFSA. We're also looking at the possibility or the opportunities by bringing in another sort of complementary to the read across that is typically based on chemical structure. So in read across, you compare one structure to another using the information from the source structure to predict the chemical and other toxicological information for the target structure. Now we also want to make use of biological read across to see when we can read across biological effects from one compound to another compound. And of course, this would be strengthened by using NAMs. So we're also exploring how NAMs can contribute to the read across. Of course, as I already said, this is under development. So these are currently ongoing work where we study what could be the contribution of these approaches. The other areas where we recently brought in the NAMs And this is something that has been ongoing for the past, let's say, 2-3 years, where we really look at opportunities to bring in NAMs when we develop guidance documents for regulatory purposes, where we see where there are gaps, we see where the technology of NAMs could be an added advantage to over animals to provide us with information that is needed for the risk assessment. And of course, primarily this is dealing with mechanistic information. So we started with looking at nanomaterials, and with the guidance that was published in 2021, so last year, just over a year ago. And we clearly explained that guidance that in vitro tests can provide insights into the hazards and mode of action of nanomaterials that cannot be gained from in vivo studies. So getting information about internal exposure, getting information about the cellular internalization and subsequent fate of the nanoparticles, getting a better understanding of the toxicokinetics and the toxicodynamics of those nanomaterials. So this is one example of a guidance document where we implementing the use of and recommending the use of NAMs. Another area where we also invested considerably in the development of or the use of NAMs is the area of developmental neurotoxicity. The problem with in vivo studies is that not only extremely costly to get that information, but it's the information provided by the in vivo study is relatively limited in the area of developmental neurotoxicity. And so a battery, a DNT in vitro testing battery has been developed, and we're working very closely with OECD on that and some partner universities. And we recently published some case studies to show the support that this in vitro battery can provide for the regulatory risk assessment. I should also mention that at EFSA, we do not have any laboratories, so any laboratory work that is being carried out and that we can report on is done by outside contractors, which are typically universities and agencies, for example, as the BFR, where we had already had a presentation from Gilbert today. We're also advocating the use of NAMs in the area of grouping for chemical risk assessments, so dealing with mixtures of chemicals, how best to address mixtures of chemicals. But one important approach is trying to put together the chemicals that are related. And this guidance that was developed really emphasizes the importance of putting or the opportunities that are presented by putting chemicals together, grouping chemicals that have a common mode of action, a common AOP. And again, to be able to understand or to be able to group them together, we really need to have better understanding of mechanistic information. And this can, again, only be provided by, or generally only be provided by using NAM approaches. So what is currently ongoing? A number of projects have been outsourced over the past, I would say, 2 years. This is just to share with you some of those projects that are being carried out outside EFSA. So we have a project on pesticides looking at neurodegenerative diseases. Another project on nanomaterials, where we're focusing on the gastrointestinal uptake of nanomaterials using NAM approaches and the genotoxicity of nanomaterials. We are looking at opportunities to use artificial intelligence to support NAMs. We also got a project on the immunotoxicity of PFAS, the polyfluorinated compounds, using NAM approaches. We have a project on essential oils. This is not for food, but for feed, for feed additives, to be more precise, and looking at interspecies differences. We have a big platform that it was developed over the past 5 years in EFSA. It's now the new version that is out. So the TK Plate 2.0, which is an open source platform for integrating PBTK models and machine learning models. And we also have a project on trying to quantify the human variability in toxicodynamics. And this is based on QAOPs. We have also a number of projects that have been, let's say, published, whether they have been published for tender specs, so people can, or laboratories can apply to carry out those projects. Some have now come to an end in terms that we're currently going through the evaluation of the application that we have received, and they will be allocated soon. We're talking here about over the coming months. So development of AOPs for developmental neurotoxicity, but more the peripheral side of neurotoxicity, framework for metabolic syndrome in the context of endocrine active substances, AOPs for stress responses and glucocorticoid production, NAM transcriptomics data to enhance the understanding and the prediction of target organ toxicity. We also have a project on having better models, developing better models for ADME, for the next generation risk assessment, and also a project on NAMs, developing NAMs approaches for nanomaterials. And just to give you, with the next slide, a bit of an overview, the way we see ourselves going. This slide is to illustrate a bit where we are and where we're going. And I would like, first of all, to start with on the right-hand side. So we have on the top, when it comes to integration of hazard identification and hazard characterization, very much based in the past on in vivo toxicity studies, but now we see what we call the NAM data cloud, where we're getting the support from new information, new data sets, both from in vitro studies as well as from in silico studies, in vivo alternatives, to help and to be integrated with the hazard identification and characterization. But we also want to see the whole two pillars that are exposure and hazard identification and characterization, a bit more overlapping. Typically, they were quite separate from each other, and you would do them completely separately, the two assessments. But there's much more of a commonality, and we want to have a much more of a holistic approach. We're really trying to understand a bit more what is the overall integral exposure from different sources, not only from consumption, but and occurrence, but also trying to understand better what is internal exposure, what is the non-dietary exposure compared to dietary exposure, and taking this exposome, how we can use epidemiological data and human biomonitoring data to integrate this information with the hazard information, trying to link the two with the right toxicokinetic data to be able to predict what will be a safe exposure level for humans in our risk characterization. And then, of course, we're not working on our own. This would be impossible. We are working with, of course, URL ECFAM that you've heard already. We are working closely with ECHA, our sister agency. We are working with a number of projects that at the European level. We have the EPAA, we have Horizon Europe project, the Big Park project partnership that has been started earlier this year. We're working with a number of other European funded projects, such as those from the ASPIS consortium and the EU Tox Risk that has now finished. We're also working at the international level, where we're engaged in APCRA, the acceleration of the pace of chemical risk assessment from EPA, OECD, Merck is our own network with other agencies throughout the world. We're working with OECD, with the WHO, FAO, the Global Coalition for Regulatory Science Research, and of course, let's not forget our national food safety agencies, both in Europe and outside Europe. And then just some final thoughts about how can we move to this next generation risk assessment for food safety. So talking a bit about visions, expectations, opportunities. Of course, there are still a lot of challenges that exist when we talk about NAMs. We need to have the confidence in using NAMs. The NAMs need to be benchmarked. It needs to be robust as a tool. We have to be sure that the coverage, the biological coverage provided by NAMs is sufficient to identify any adversity that you would see in an animal study. We need to better understand how to use the data, how to interpret the hazard data coming from NAMs, how to use them in terms of protection or versus prediction, how to integrate them with your traditional data. We need to, of course, have the right training, and that is also at the European level very much emphasized. We need to train the scientists on how to use NAMs in a regulatory context, but also the risk assessors, how to understand and use NAMs for the regulatory work. And this requires a lot of efforts from all sides, and it's important really to say it involves a collaboration among all actors to be able to achieve this. The regulators need to work closely with academia, with society. There needs to be involvement of industry, of course, in the development of the products. And of course, these resources, these efforts need resources, and of course they need funding, which is very important to really to be able to bring us into the next generation risk assessment using NAM-based tools for regulatory purposes. And with this, I stop my presentation, and I thank you for your attention.
Thank you, Dr. Georgikos. You have given us a comprehensive overview about the EFSA's roadmap on NAMs. Thank you very much. Any questions and comments from the audience? Okay, thank you, Dr. Georgikos.
Thank you.
We will maybe connect with you and privately. Thank you so much.
My pleasure. Thank you very much for the invitation.
Thank you again. Dr. Jia.
OK，
please.
好，下一个报告啊，我们下一个报告呢，报告人是郭嘉斌博士，他是嘉宾博士呢，他毕业于北京大学，获理学博士学位。2007年到09年呢，在美国加州大学尔湾分校留学，从事毒性测试与替代方法研究已经20年了，承担一些国家的重点研发计划等多项国家和军队的课题，发表了学术论文有70余篇，主编或者参编专著呢12部。他还兼任中国环境诱变剂学会毒性测试与替代方法专委会副专委员等职务。他今天报告的题目呢是基于细胞的体外毒性测试整合评估在食品安全风险评估中的应用，也是因为疫情的原因，只能通过线上做报告。好，下面呢，有请嘉宾博士。
好，谢谢贾老师介绍，也特别感谢咱们组委会贾老师的邀请，来有机会给大家一起分享我们做的一些工作。这个共享一下题目啊。今天我给大家一起来交流的主题是从这个体外的测试来开展这食品风险评估中一些应用，包括一些考虑的因素。那么我们都知道，大概15年之前，在美国的国家研究委员会 提出了在毒理学领域是具有一个里程碑意义的一个报告，那就是这个毒性测试21世纪的毒性测试策略远景与展望。那么在这个测试当中，报告中呢就突出强调，这个在未来的毒性测试应该要更多的从动物实验，主要以动物实验为主的这些测试方法来转移到以基于这个人源细胞和人源细胞组分一些测试的高通量的测试方法，当然也包括一些像刚刚唐老师所介绍的一些计算毒理学的技术方法。在2013年 OECD 呢，也颁布了这个关于有害路径 LP 的这个指南。其实这个指南跟 TDRCC 有是一个很多的是一致的。这个从早期的分子事件到整个关键的细胞反应 Key Event， 以及关联到最后的有害结局，强调毒性通路的测试以及一些体外的和新技术新方法 NAMs 应用在咱们风险评估中，它的重要的作用。那么在 TDRCC 当时也是勾画出咱们这个整个毒性测试所用的一些选择，包括从当时这个主要依赖动物的传统测试方法，针对这些传统测试方法所存在的一些缺陷，包括这个剂量比较高，通量比较低，而且时间比较耗时。那么通过这些毒性测试技术方法，预计这个比较理想的，或者在未来我们期望的是要从这个动物的测试方法转移到主要依赖于人源动物学的，就是人源细胞和细胞组分相关一些测试方法，而且测试剂量应该涵盖一个更广泛的剂量范围，从一个这个更加高通量的测试角度，更加这个节约时间来开展相关毒性测试，而且这种毒性测试方法应该高度帮助一些关键的细胞反应、关键的毒性通路的改变，而且尽可能的采用一些体外测试方法和计算毒理学方法，这也是咱们未来毒理学发展的一个重要方向。那么这个从2007年的 TDRCC 提出来，应该说在毒理学领域是收获反响是非常大的，而且这个引起了全世界的关注。那么大概10几年过去了，其实在这个领域的发展也是非常快的。包括这个2017年美国 FDA 也是进一步结合美国 FDA 的职能，在这个药物评审，包括食品这个评审当中，进一步提出在整个评审过程中应该加快对这些毒理学新技术新方法的一些采纳，包括这个新技术新方法所获得的数据如何整合到这个整个评估当中。在这个 FDA 这个预测毒理学这个 roadmap 还特别强调着这个风险评估，包括咱们毒性测试，应该从动物实验，然后更多的要拓展到从不同层次的生物学层面，包括从基因层面、蛋白层面，以及生物学通路层面和细胞器官功能层面等等，通过这些不同层面的生物学的反应来综合的评估外源性物质，包括药品也好，还是化妆品还是食品等等对人体健康风险的潜在的不良影响。那么在这个 roadmap 还突出强调几个这个非常有发展前景的一些这个技术，其中包括微生理系统，也就是器官芯片，以及这个在生殖发育毒性当中的替代方法和计算毒理学方法，包括这个体外的这个替代方法，以及 QSA 和 Red Cross。那么在2019年，美国这个 EPA 也是做了一个非常重磅的一个宣布，就是计划在未来的这个时间要大幅减少这个化学物评审过程中对传统动物实验的依赖，预期是希望能在2035年之前，几乎要禁止这个哺乳动物实验来开展相关的化学品质测试。当然，这个宣布或者作为决定，在业内还是影响非常大的反响，有存在一些争议，但至少也说明代表在整个咱们毒性测试一个非常非常重要的方向，就是越来越多的要采用这种非动物的测试方法来开展相关风险评估。那么在2021年呢，美国 FDA 又进一步在他们的管理学前沿当中也提出这种非动物的测试方法在临床前，也就是非临床评价中要进一步的采用这些非动物测试方法，也就是咱们所说的这个新技术新方法 NAMs 来减少和优化或者替代这个传统的一些动物实验。那么今年在9月29号，这个也是一个非常大的一个事件，就是美国这个已经通过了这个现代法案2.0。这个在这个法案当中，美国 FDA 又进一步这个明确，要在整个药物的非临床评价当中，要大幅的减少对动物实验的依赖，尽可能采用一些非动物的测试方法，采用一些新的技术方法来开展这个药物的非临床评价。 那么在2021年呢 ，OECD 也同时也颁布了一个，应该是全球世界上第一个不采用动物实验开展一些毒性测试的一个指南。中国应该说这些年这个整个对这种新方法、毒性测试新方法的发展也是非常重视，而且发展也是非常快的。尤其是这些年，自从这个2014年，我们在中国毒理学会和中国环境用品学会也成立了这个毒性替代方法这样彭航青老师的带领下，也成立了相关的专业委员会，组织了全国的相关的专家，开展了这个大量的相关的研究。那么从法规层面，特别值得这个关注的一个，就是在今年的3月份，中央人民政府和国务院办公厅，这应该来说是从中国政府层面非常高的层次，提出了关于加强科技伦理治理的意见。那么在这个意见当中，就非常明确的这个提出这个科技伦理的原则，在第2条要尊重生命，里面就明确指出，在使用动物的时候，应该要符合3R原则，即减少、替代和优化的要求。这应该是中国政府非常高的层面上首次这么明确的提出3R原则的要求。具体到咱们这个化学物的登记，在不同部门，比如说这个生态环境部针对这个新化学物质登记的指南，这是由生态环境部来主要承担管理职能的部门。其实在2020年。这个第51号就已经这个发布了新化学物登记指南。那么在这个指南当中也是非常明确的，提出一般的原则，就是新化学物登记的时候开展的毒性测试，就应该在保证风险评估质量的前提下，尽可能要采用这个3R符合3R原则的一些测试方法。那么具体的毒性测试方法而言，其中这指南就提出应该采用化学品测试导则。其中这个化学品测试导则里面就有很多一些体外的方法，一些非动的测试方法也被收录其中。特别是在里面还提出，这个如果是中国境内开展的毒性测试或者风险评估的方法，也可以采用咱们 OECD 已经颁布一些指南。如果这个中国的化学品的测试导则或者中国的相关标准还没有及时更新，而 OECD 的这个测试指南或者指导原则已经更新了，那么在这种情况下，如果我们采用 OECD 最新的指导原则开展相关的毒性测试和风险评估，也是可以接受的。如果是境外开展这些实验，那么基本上按照这个 OECD 颁布的指南，这个从化学物质登记的角度，这些技术方法应该还是都是能得到认可，或者采用这个其他广泛认可的一些方法开展毒性测试，包括咱们非动物的测试方法和体外的方法都是可以得到应用的。那么其实在我们食品安全领域，这些年也是发展也是非常迅速。包括咱们这个国家食品风险评估贾旭东老师团队，也是这些年开展了非常好的一些工作，包括在推动这个体外方法和这个计算毒理学方法一些非动物测试方法在食品风险评估应用也是开展了大量的工作。包括这个我们贾老师他领导的这个食品这个计划毒理学计划，这个也是开展的针对一些新方法也做了大量的工作。那么从体外模型而言，在毒性测试咱们所采用的常规的或者比较常用的模型，主要包括一些原代的细胞，包括细胞系以及这个干细胞。特别是近年来非常这个受到这个推崇的，就是人源多囊诱导干细胞也是越来越多应用到毒性测试。从事不同的模型，还有这个咱们3D 培养的细胞模型，包括通过 bioprinting 的这个 cell， 就是3D 的生物打印模型，这也是越来越受到关注和运用的。此外，还有这个近年来也做的比较多的
as well as the organ on chip, which has been used more frequently to risk assessment. And under the leadership of Peng Ganjing, and he was one of the pioneers to develop the toxicity testing. And he has also undertaken a lot of national key projects. And the NAM, especially the in vitro toxicity testing, and that can be widely used in the risk assessment of food. And from this perspective, and we focus on some targeted organs, and as well as the And for such a targeted organs, and we set some in vitro models. And through the in vitro testing, and we assess we will set up the pathway. And so there are different cells, and including the human cell 3D culture, iPSCs, organ on chip, NARF2, GSH, etc. And then the models are set up for high content analysis, high throughput screening. And so in combination of the in vitro assays, NAMs, together with some clinical documents, and we started the combined integrated risk assessment. And as mentioned by Dr. Fang, PPDK model, and it can contribute to the in vivo to in vitro extrapolation. And then it enables us to do some human health risk assessment. And I will show you some case studies. And firstly, about the toxicity mechanism on the targeted organs, for example, liver and heart. And we needed to select some human cells. And firstly, we need to know exactly about the characteristics of the cells, its advantages and advantages, pros and cons, and to what extent it can represent the characteristics of the human body. And for example, the hepat cell, hepatocytes. And another focus is the quick screening, and we pay much attention to the active oxygen, and it is related to the cell death. And based on such common active response, and including the antioxidant and the biogenesis out of G, and it can help us for cell screening, and we can also check different parameters at the level of cellular functions, and to monitor the chemical influence on the targeted organs through in vitro approaches. And we know that the liver is a key organ for metabolism, and we culture different cells, and for example, they have the hepatocytes and HepG2, LO2, and HepRG cells, and HepRG cell, and from different perspectives of the metabolic enzyme, and as well as the BPA, and which may lead to the high liver toxicity if used overdose or during overdose using. And we also see that the activity of the enzyme is different. And we also use different approaches, and especially the 3D culture, the HepRG cells, and to check the metabolic ability, and then the Hepar cells, the HepRG cells can better characterize the human cells. And we see that we made a comparison between the 3D cultured hyper-RG cells and another kind of cell that can further validate the hyper-RG model. Is very practical and significant, and in toxicity assessment, and because for hyper-RG cells and they are different toxins, and through the toxin testing, and we get the PBD value. And for human-induced pluripotent stem cells, and we have the derived cardio myocytes, and because for some AC16 96 16 cell can characterize the human cells, however, and some cells are fluctuating, but they HPSC SCM cells, and we can see through the literature study, and we can see such cells and can better characterize the biological characteristics of human cells. So the human induced pluripotent stem cells can better characterize the cells on the human cells, and with application of HPSC CMs in assessing cardiotoxicity, and it has a different advantages, no matter in terms of the characterization, in vitro toxicity characterization or other aspects, and it has advantages. And if we use such cells to assess the nanomaterial cellular toxicity, as well as its impact on the cellular functions, and we can sensitively capture the influence of some chemicals on the cells, and we further assess the damage to cardio myocytes, and moreover, and we had a tool and for screen the early cells, and we through the genome, and to screen and some toxins, and we screen some potential biomarkers and biological pathways by toxic omics, and we got really useful information for further development. And through the screening, and especially the cellular stress pathway and the adverse outcome, and it can provide a very valuable information to the risk assessment. And we know that the chemical enters a human body and has a series of reactions with the cells, and it can modify the cells. So from the contact between the chemicals and the cells, and to the transducers or transcription factor, or the cell injury, and even the cell death. And we can use this perturbation and to show such a process. And we know that there have been some rules to follow, or some characteristics for the modification, and maybe the in early times the cellular response was not that strong. It is quite healthy to the body, but through the cellular oxidative defenses, and it may have some adverse changes, modifications to the cells, but there is no obvious cell injury. or some reverse changes. However, and if the exposure time is longer, and it will lead to the unreversible modification in the cells, and then the toxicity may also change in the process. And so it is very key to us how to identify on which POD or under which dose, and it will cause the irreversible cell injury.
A very important pathway we pay attention to is PGC1 alpha pathway, which has been proven to play a very important role in regulating cellular stress. So by building the PGC1 alpha pathway, first of all, we use some in vitro method to review the impact on the pathway, and from the cell level, protein level, we review the patterns of the pathway, and then we use a computational toxic mode of toxicity pathway to use in vitro data and combine it with toxicity pathway to analyze the potential pattern and characteristics. For example, identifying the toxicity response value so as to achieve the integrated analysis of in vitro data. And another pathway is NRF2 pathway, which is mediated regulation of cellular stress response. OECD 442D has also included NRF2 pathway as a very important pathways, including in autopathy, inflammation activation, and stress and UPI, as well as mitochondrial redox signaling. And as for skin cell injury, use that pathway as a key for a test to observe the change of the pathway. And during the test, we have used a normal cell mode and a special mode with alpha knocked out, so as to observe the mitophagy in skin cell injury and see the role of an NRF2 pathway in skin cell injury. And we have also reviewed some toxicity dose characteristics. And so traditional methods actually has the low throughput and the time is quite time consuming. So we used SPR method to detect the pathway change. So there are two molecules here, two important molecules here, which are more sensitive with a higher throughput to detect the change of the pathways. So you can see the results, which have been further validated to see the alignment. And we have also applied a national patent. And the third part is to integrate in vitro data and the PBPK modeling, AOP, so that we can extrapolate the in vitro data into human body. And you can see the change of the pathways and the establishment of the PBPK model. And we have done some work in hepatic cells, and we use PBPK to predict the hepatic toxicity or TR toxicity, combining with in vitro data to build this in vitro to in vivo extrapolation by PBPK, so as to see the important toxicity parameters and dosage for obvious hepatotoxicity change. And so when we build the models, we have used a 3D model to verify the data of the 3D model, and then extrapolate the human risk or hazard, and compared with and combining with clinical data, it's easier to be validated or to obtain successful models. And we found the toxicity data is quite successful in predicting the hepatotoxicity. and the paper has already been published. And finally, a summary. What are the considerations we need to have? So first is from the origin. We need to select the right in vitro models. So to simply put, it needs to be able to characterize the real situation of the human body. So the origin, the target organs, and the TP21C also mentioned that we need to use human cell, and also we need to have target organ, and if possible, we can also use the human primary culture cell. Or some very good cell models. For example, for toxicity HepA-G cell, in the toxicity test, proves to be a very useful model. And also iPSCs. So selecting the right vitro model, and we have also used 3D culture. Of course, a 3D cell can better simulate the real situation of the human body because the human body is a three-dimensional structure. And when we select cell lines, we also look at the characterization of the cell, the health status of confluence, passage, and age, etc. So on the one hand, we need to discover from experiments, and we can also refer to some good literature. Second is exposure scenario. We need to be able to cover proper health scope, and selected dosage needs to be relevant to in vivo exposure. Sometimes in order to change the toxicity, the dose is adjusted. So it is very different from a real exposure. And as for the exposure time or duration, whether 24 or 48 hours, that also need to be taken into consideration. Oh, and we also need to consider whether it's a single or repeated treatment, and the low dose effects. The third level is chemicals and metabolism. Including the chemical physical property of tested substance and its distribution. For example, some chemicals will not go through the blood-brain area. And the fifth is identifying the toxicity pathways and the key events. And the relevant, and next one is relevant biomarkers and endpoints. The endpoints can be sensible, reliable, and relevant. And sometimes we need to adopt a combination of methods or an assay panel. And we also need extrapolating from in vitro data to in vivo effects, and also whether GI-VIMP and the GCCP compliance also need to be taken into consideration, and also the validation and regulatory acceptance and uncertainty need to be taken into consideration, and also the uncertainty, feasibility, operability, simplicity, ethic, time consumed, and the economy of the test also need to be taken into consideration. Okay, that's all for my presentation. Thank you, Dr. Peng, for his guidance. And also acknowledgement to our team and also partners, including Unilever and experts from US Emory University and Beijing University of Medicine, Capital University of Medicine, as well as the relevant funding support in China. So starting from 2009, these are some of the publications made by our team. And just now, Dr. Cass has also mentioned some publication which we have also been engaged. If you're interested, please contact us. Thank you.
好，谢谢嘉宾博士。看看现场和线上大家有问题吧？
Any question?
彭教授，您那边有问题吗？现场？
这里有一个问题。
Thank you, Professor Guo, for your wonderful presentation. I have a question. Your report, you did not pay much attention to the hybrid or combined exposure. So can you explain the in vitro? So in terms of in vitro test, what should we take into what should we consider for combined exposure of compounds? That's a very good question and quite tricky. As for the combined exposure, for example, like the traditional Chinese medicine or some other materials, actually you're not very familiar. You do not understand quite the ingredients and also the content or the concentration in those materials. So as for in vitro study, on the one hand, we need to ensure that the compound's quality standard of every batch should be aligned or similar. For example, for TCM, among different batches, because of quality assurance, the result of the study may also vary a lot. And second, from toxicity test perspective, we usually look at the toxicity effect. For example, to which extent it can, say, kill 50 or 70% of the cells, and then combine it with the characteristics of the compound to make a comprehensive study. Thank you. Okay, thank you. In the interest of time, thank you. That's all. Next. Professor Peng. Okay, the next speaker is the research assistant professor from China National Center for Food Safety Assessment, and Dr. Xi Miaoying, and her topic is a new approach and methodology, NAM, for the prediction of chemical-induced cardiotoxicity in humans. Dr. Xi Miaoying joined the China National Center for Food Safety Risk Assessment in 2021 after she obtained her PhD in toxicology from Wageningen University in Research in the Netherlands. She is registered as a European registered toxicologist and is currently a research assistant professor in the toxicology lab of CIFSQ. Her research interests include the in vitro-based chemical safety evaluation, hazard assessment, physiologically based toxicokinetic modeling, alternatives to animal testing. Currently, she has focused on integrating in vitro data and PBTK modeling into food safety hazard assessment. The stage is yours, Dr. Shi. Okay. Thank you, Dr. Peng, Professor Peng. And thank you for giving me this opportunity to share our research progress. So next, I will use several cases to demonstrate how we apply NAM into compound hazard analysis and risk assessment. So my topic is a new approach and methodology for the prediction of chemical-induced cardiotoxicity in humans. So these are the contents for my report. First is background information, including the development of NAMs and the introduction of terminology. And second, I will use methadone as an example, methadone and ibogaine as an example to show how to demonstrate how we use NAMs. And the last part is a summary, is outlook for the future work. So first of all, I'd like to talk about the necessity of developing NAMs. So as experts have already mentioned, in conventional toxicology, the hazard of evaluation and risk assessment mainly rely on animal studies. We know that the extract rate, that effect on human body from animal-based model has some its limitations because of the difference in species. As a result, the animal model may not have very high relevance with the human body, and with more awareness of environmental protection and higher awareness of health. So different regulators have higher demand or more restricted demand on compound safety. For example, in Europe, starting from 2006, as for industrial compound, food additives, and cosmetics, there are relevant regulations and legislations rolling out, which requires a detailed toxicology data for compounds. So in order to meet those crazy demand on toxicology, then a lot of animals need to be used for safety assessment. And we know that animal experiments need costly and time-consuming, and there is also ethical issues.
And so the REACH regulations and the cosmetic regulations have strongly encouraged the animal testing alternatives so as to avoid repeated overlapped animal testing, and to encourage NAMs. And in the new EU directive on the protection of laboratory animals should be committed to developing and verifying alternative methods. And so in terms of the limitations in the economics, and it is more and more important to find the alternative methods for animal testing. And we see that in recent 10 years, researchers in academic and industrial circles have devoted themselves to developing new testing methods for hazard assessment and safety assessment of various chemicals. And so the new approach methodology or non-animal methods have been proposed as a terminology. And in the broad sense, NAMs refers to any testing method that can significantly reduce or avoid the use of experimental animals. And we see that relevant research and management institutions in various countries and regions have included NAMs in their development blueprints and technical guidelines. And the EPA put forward the TOX 21 and TOXcast programs encouraging people to use in vitro models based on human biomaterials and animal experiment substitution methods. And ICCVM of the United States emphasized the importance of developing alternative methods for animal experiments in the strategic development blueprint for establishing new methods for evaluating the safety of chemicals and medical products, and integrated the new technologies such as organ-like omics and computer models into alternative methods. And in the work plan of new technologies and methods of EPA, and it has put the NAMs on the top priority and to promote its development and applications. And they have also formulated short-term and long-term goals and strategies to reduce the use of vertebrates for toxicity testing. And the EU Horizon 2020 plans to significantly increase scientific research investment in animal experiment alternatives and promote its application in compound safety prediction and risk assessment. And as mentioned by Dr. Park, the chemical strategy for sustainable development of a non-toxic environment issued by European Commission, and strategy 2027 issued by EFSA has been promoting the development of NAMs based on animal experiments method and improve the quality, efficiency, and speed of hazard risk assessment of chemicals of food-related substances. And in recent years, and the NAM is developing really fast in China. And as mentioned by Professor Guo, and from the management and research institute levels, and everybody is vigorously promoting the application of NAMs. And usually, in vitro models are used to screen the toxicity of compounds, obtain the dose-effect relationship, and study the potential toxicity mechanism. However, compared with in vivo experiments, in vitro models don't take into account the toxicokinetics of compounds. For example, and the process of ADME, absorption, distribution, metabolism and excretion. And so the in vitro data can't accurately reflect the exposure of compounds. And so it cannot be directly used for risk assessment, and because it cannot reflect the real exposure of compounds in vivo. So it is necessary to integrate the toxicokinetics data and related the toxic effect to the external exposure dose level of human body. And so it can be realized by reverse dose method based on PBK model. The basic principle of this extrapolation method is to assume that substances have the same qualitative and quantitative toxic effects at the same cell exposure concentration in vitro and in vivo. And the general flow of reverse dose method firstly, the dose effect relationship of compounds based on a certain toxicity endpoint was obtained by in vitro model. And secondly, the compound is modeled by PBK to simulate its ADME. Through PBK model, the internal exposure concentration and the external exposure dose of the compound can be related. And in the reverse dose method, the toxic effect concentration of in vitro test is a substitute concentration that may cause harmful effects in tissues or organs in vivo. Then we can assume that the effect concentration obtained by in vitro model is equal to the exposure concentration in human body. And then the in vitro concentration response curve can be quantitatively transformed into the external exposure dose response curve, and then the point of departure, such as the benchmark dose, can be calculated, and so as to provide a better, more information for risk assessment. And we know that the importance of the electrophysiological cardiotoxicity and the main reason of failures during drug development, and it is also some plant alkaloids show potential cardiotoxicity, and currently the assessment is not enough. And for example, and taking the methadone and ibuprofen as model substances, and it has a certain cardiotoxicity, however, and the those effect relationship is not clear. And so, we can use the PBK targeted model and to establish the substitution method. And firstly, we should choose an appropriate in vitro model and to generate the in vitro toxicity data. And so we selected 11 substances and to evaluate the effect. And firstly, and we choose the mice stem cells, and we also detect the inhibition of such cells. And secondly, and we choose the different cells, and we select some electrophysiological parameters. And in terms of the criteria for the selection, and Mr. Xia also mentioned that we need some criteria for reference. And firstly, we will check whether these two models are sensitive. And we can see the different positives, the concentration curves. And we can see the black color refers to the data from the mice body, and through comparison, the black is the mouse cell model, and the green refers to a human cell model. And we can see that the human cell is more sensitive. And in this graph, and we can see green. So refers to that the effect concentration that the positives arouse 10% in the serum concentration, and after comparison, and we can see for most of the positives and the correlation between the in vitro and the in vivo data is quite good, and so we can see that it can be used as the basis for in vitro in silo approach, and we chose the human cardio cell cardio myocyte as this, and then I will take the methadone and ibogaine as the model substances, and to show how to construct a reverse dose method based on PPK and verifies its accuracy in predicting human cardiac toxicity. And in the in vitro model, the toxic effect of the model substance is reflected by the change of electrophysiological parameters. And we know that as shown in the figure, the methadone and the main metabolite EDDP can prolong the field potential duration of myocardial cells, and the toxic effect of methadone is slightly greater than that of EDDP, and through and to predict the cardiac toxicity of methadone, and we use the reverse dose method, and we check the cardiac toxicity of the two substances, and we choose the of a few potential duration, and to show the time between the nano iron peak, and so it is the QT7 representing the QT the QT prolongation, and it is related to related to the sudden death, and so it is a very important parameter and we can see that methadone and EDDP obviously induce the cardiotoxicity in a human cell model, and the toxicity of methadone is slightly bigger than that of EDDP, and so we have the analysis of the in vitro in silico approach for methadone, and it is helpful for us to judge whether we should consider the toxicity of the metabolites. And next, and we should establish the PBK model, and we describe the different cell lines and different forms, and to describe the ADME of compounds in human body, and we need two kinds of parameters, and the physiological, and for example the organ volumes, blood flow rate, protein abundance, and another kind of parameters are compound parameters, including absorption, distribution, metabolism, excretion, and the renal clearance, etc. And we link the exposure doses to internal concentrations through the PBK model. And this is called the positive meteorological method. But if we use the internal concentrations to induce the external exposure, it is called the reverse dose method. And we can see that through the toxicokinetics data, and after comparison of the data, and we can judge the accuracy of the model. And we established the reverse dosimetry, and the two paragraph on the shows the predicted data, and the points are reported in have been reported in some literature, and after comparison we can see that the overlapping of the data is very high, and the deviation is just a 1.5-fold difference.
And when we extrapolate based on the assumption that, as mentioned before, that the in vitro test toxic effect concentration is a surrogate concentration for the deleterious effects of the tissue in vivo. Therefore, when we do extrapolation, it follows that free in vitro effect concentration equals free internally exposed in vivo exposed maximum cardiac venous blood concentration, since the latter can be extrapolated to the in vitro exposure dose by PBPK model, thus correlating the in vitro concentration with the in vitro exposure dose. Applying this step to all in vitro effector concentrations, then the entire in vitro concentration response curve can be converted to in vivo dose response curve, and then we repeat that to convert that curve. One thing to mention that although EDTP presents a similar toxic potency to methadone in the in vitro model, EDTP blood concentrations are much smaller than its in vitro toxic effect concentrations after administration of clinical doses of methadone. So therefore, the toxicity of EDTP was not considered for extrapolation. The last step is validate the model prediction result, which is the curve of in vitro exposure and response. So the graph shows, you can see in the graph that the line represents a model prediction result, since because of the larger individual differences in plasma-free coefficients of methadone. So the extrapolation took into account the plasma-free coefficients of populations with different physiological characteristics, and 4 dose response curves were obtained. And these represent clinical data and human data. And by comparison, you can see that the overlapping is quite good. So which means we use in vitro cardiac toxicity data and combine it with PBK model, we can predict the methadone-induced cardiac toxicity. And next is an example of ibogaine. Ibogaine is an alkaloid extracted from the root bark of the African plant ibogaine, which is widely used in some West and Central African countries for local shamanic religious rituals because of its strong hallucinogenic effects. Both ibogaine and its metabolite are neuroactive, and several human and animal studies have shown that single dosage of ibogaine and d-ibogaine are effective in reducing drug withdrawal reactions. However, the safety of ibogaine and d-ibogaine in the treatment of drug addiction has been controversial, with some patients experiencing side effects such as prolonged QT intervals, cardiac arrhythmias, and even death after taking high doses of ibogaine. And clinical trials have also shown that non-ibogaine can cause dose-dependent interval prolongation in drug-addicted patients. So first of all, we use the cardiac cell model to detect the cardio toxicity. And so here you can see that it can prolong the dose reliance. And ibogane's toxicity is a little higher than noribogane. And next for ibogane and noribogane, we established the PK model and compared the vitro and in vivo data to see the overlapping both for ibogane and noribogane. So which means our model has been validated and can be used for later application. When we extrapolate the toxicity of ibogane, what is different from methadone is while taking clinically relevant ibogane, then the noribogane dose concentration actually is higher than that concentration in vitro model. So which means when we can look at the ibogane concentration, we also need to take that of noribogane into consideration. And here we used the equivalent method, and here we still need to. So you can see the equation here, this which is the assumption we use. But the maximum concentration here is calculated or after the corrected TF. So the toxicity equivalence factor TEQ can be obtained from cardio cell. And considering that the direct administration of noribogane can also cause a cardiotoxicity in humans, the inverse dose method was used to predict the dose QTC interval prolongation relationship for noribogane in vitro exposure. And here, in order to further explore the magnitude of the contribution of prototypes and metabolites to prototype-induced human cardiotoxicity, was compared between the free maximum cardiac venous blood concentrations of TEF corrected ibogane and noribogane in humans after administration of different doses of ibogane. So from this graph, you see that based on the ibogane concentration toxicity contribution, the toxicity of noribogane actually is higher than that of ibogane. And finally, we need to validate the model for in vitro exposure to see whether the curve predicted is accurate or not. So on the so the relationship between in vitro exposure dose and QT interval prolongation for ibogane and noribogane are shown in the left and right panels respectively. And the comparison shows that the predicted results and the in vivo data largely overlap. And in order to further quantitatively verify the accuracy of the model predictions, BMD analysis was performed on the predicted and literature reported dose response curves for noribogane, ibogane in vitro exposure, and the BMDL values differed very little, basically within 1.5 times. So which further reflect that our model has very good prediction result. By comparing the BMDL values of ibogane and noribogane, it was found that both are relatively similar, which means that both prolonged the QT interval with similar potency. And combined with the proportional contribution of both to the toxic equivalent concentration, it was hypothesized that it was noribogane rather than ibogane itself that played the dominant role in ibogane-induced cardiotoxicity in vivo. From this case, we can show that the integration of the TEQ method into the inverse dose method, as demonstrated by the example of ibogane, allows the prediction of cardiotoxicity of chemicals with active metabolites. And finally, a summary of our study. By comparing ibogaine and methadone, so we find that the findings provide insight into the factors that influence the relevance of cardiotoxicity of metabolites in the reverse dosimetry for the prediction of parent chemical-induced cardiotoxicity. Second, PPK-based reverse dosimetry of in vitro cardiotoxicity data provided adequate predictions for human cardiotoxicity of methadone, ibogaine, and noribogaine. And third, the developed approach can be thought of as a proof of principle for the prediction of cardiotoxicity in humans, thereby contributing to non-animal-based nets for hazard assessment and safety evaluation of chemicals. Here I've shown the basic applications for basic applications of reverse dosimetry, but when we combine it with Macaro and other dose models, then we can use that to predict the individual difference of compounds in toxicokinetics or its toxic effect in specific population, and also they combined to predict the combined exposure of compounds. And our next plan is to apply the reverse dosimetry into safety-related safety assessment and use quantitative method to improve accuracy. And now we are having a project which is using reverse dosimetry to predict salads and its impact on human lipid metabolism, and we hope there are opportunities for us in the future to share such research. Thank you again for all those who have contributed to this research, and thank you to all the audience online. Thank you for your patience. I don't know, maybe there is no time for a question. If you have any advice or question, welcome to contact us. Thank you. Okay, thank you, Dr. Shi, for your wonderful presentation. It's quite comprehensive in introducing the in vivo, in vitro extrapolation and the specific procedures and principles, particularly based on some case studies and the application in cardiotoxicity in humans. It's very excellent, but in the interest of time, we are already far behind the schedule, so there is no time for further discussion. Thank you again for your report. Okay, the next speaker, sorry, Professor Jia will moderate the next presentation. Okay, thank you, Dr. Peng. The next is our last presentation. The speaker is Suzanne Fitzpatrick from the US. And Suzanne is the Senior Advisor for Toxicology, Center for Food Safety and Applied Nutrition, US FDA. And she is a board-certified toxicologist in the US and in Europe. She chairs the FDA's Alternative Methods Work Group that is currently focused on in vitro microphysiological systems. She helped to develop the FDA DARPA and CATS program on organs on a chip, and continues to work and give presentations on this evolving area. And I know it's quite early here in the US, and she will join us online. And the topic is advancing new alternative methods.
So can you hear me?
Yes, I can hear you.
Can you hear me? Thank you very much for inviting me to speak today. Closer to the microphone. And I'm wondering if you can hear me talk.
Yeah, yes, we can hear you.
Okay, and you have my slides? Can you show my slides?
No.
You have the, yeah, you have the slides, and when we did the practice, we went through them.
No, I can't see your slide.
Yeah.
Could you share?
You have the slides.
Oh, you want us to show the slide?
Show them, yes.
Okay.
I want you guys to show them. That's what we agreed upon.
Okay, 那工作组能帮了一个帮了 分享一下吗？
That's it.
Okay, wait a moment.
Okay.
贾教授他能看见吗？你问他一下。
看不见，我这边就看不见。
So, can you see the slides?
这边只是显示了一
No,
个
I can't see them.
没有连上。
Can you see the slides? Because we have shown the slides. Sorry, maybe it is shown on the screen. However, it hasn't been shown in the Zoom meeting.
不好意思啊，请大家稍微耐心的等一等啊。
Sorry, some technical problem, and we
Okay.
need to wait for a minute.
这样共享一下这个。来，第一个吧。OK。这两个。现在可以了吗？
Does it
here we
work?
are. Okay. Yes, it worked. Hooray.
Thank you.
Okay, so I'll just say next slide when I want the next slide. Okay. So thank you very much for inviting me to talk to you today on what we're doing in the U.S. Food Administration on advancing new approach methods across the agency. So next slide. Okay. FDA, the US FDA has a very long commitment to promoting the development and use of these new technologies. For those of you that don't know the structure of the US FDA, we have 6 product centers: foods, where I am, human and veterinary drugs, tobacco, medical devices, and biologics. And we have very different mandates, very different products, but we are working together as one agency to advance new alternative methods. And we're working both with our regulators, which I am, and our researchers to make sure that everything we do advances new alternative methods. So next slide, please. So several things happened in the US that made this more and more important to the regulators. We had the TOX 21 consortium, which is a consortium between 4 federal agencies: EPA, FDA, NCATS, and NIEHS. We partnered, FDA partnered with DARPA, which is a defense authorization research group, to develop the first organs on a chip for use in risk assessment. We developed our predictive tox roadmap, and then we put together an alternative method committee, and I'll talk to you about each of these. Next slide. These 3 roadmaps, you've seen one of them already from ICFAM, came out at the same time within a few weeks of each other in 2017 and the early 2018. And they all emphasize what the US calls the 3 C's: collaboration, communication, and commitment. We collaborate across the federal agencies and with our global partners. We communicate what we're doing to all of our stakeholders, and we are committed to moving forward together. Next slide. This is FDA's framework for incorporating new methods. This is our predictive tox roadmap. It had 3 steps, which was working together across the agency, training FDA regulators and researchers in new approach methods, continual communication with our stakeholders, collaboration outside, making sure all of our research was to answer regulatory questions and to make sure we have oversight at the very highest part of FDA in the Office of the Commissioner. The next slide. And here's a little bit more about each of those spots. We wanted to foster communication across the different product centers and use this to leverage our resources to advance predictive talks. Next slide. We wanted to make sure that the first time our regulators saw a new approach method, it wasn't in an application. So we wanted to make sure that we continually educated them on new predictive methods, and so they weren't surprised. Or our researchers were not surprised when they saw something they didn't know what to do with it. Next slide. We want to collaborate with stakeholders like yourself, and you'll see in a few slides how we're collaborating with China through our NormAccurate Association. We felt that collaborations across disciplines nationally and internationally was pivotal to helping FDA maintain its momentum to deliver new predictive toxicology methods to make our products safer on the marketplace. Next slide. And we wanted to reaffirm our commitment to incorporate data into new toxicology methods by different communication strategies to make sure people really knew what we were doing and can encourage people then to come talk to us and to submit new methods into the regulatory process. And last slide, next slide. And we wanted to make sure we were leveraging our research so that it started with a regulatory question. We didn't want to develop new approach methods and then try to fit them into a regulatory program, because that's often very difficult. We wanted to start with a regulatory gap and then support both intramural and extramural research. And then lastly, and this is the next slide, we wanted to make sure we were tracked at the very highest level at the Office of the Commissioner, and we are tracked there, that we across the agency are responsible to the Office of the Commissioner for ensuring we work towards developing new approach methods. Next slide. So one of the things to remember is regulators had to be included upfront in any new method development. New methods, new toxicology methods must answer regulatory questions. Regulators, let me move this over. Regulators should delineate, but regulators need to start out by delineating what gaps they have, what they need, where they need additional research, and we need to have continual training for regulators to make sure that they understand what these new methods are and how to prove them when they see them in an application. Next slide. One of the things that we really pushed that started at an FDA, but now has gone across all the federal agencies through the new ICBAM validation document that's about to come out, is that we should start with a regulatory question or context of use. What question needs to be answered and for what purpose? And that really decides how much validation, or we call it qualification, is needed for a particular assay in order for someone to have confidence in using that assay to answer a regulatory question. If it's a screen, a discovery, then not as much data is needed to qualify it. If it's replacement for a pivotal study, you're going to need a lot more data in order to have confidence in that method to use in lieu of an animal study. So what the context of use, which is the very hardest thing to do in research, is, or when you're developing a new method, to answer what a context of use is, helps define the applicability domain, what you want to, how much qualification data you need. And the other point is any method that's qualified, and once we qualify a method, we make that public, it can be other contexts of uses can be added at a later date, or you can always use additional methods for additional endpoints, but they're just not qualified and you have to put in the underlying data. So it's a very flexible program. That's what we're encouraging in the US through our ICBAM, flexible, independent review, and information transparency. Next slide. So we wanted to be a little bit more, we talked to our stakeholders and we wanted to be a little bit more, we wanted to give some people a place to come to actually if they had alternative methods. So we developed the alternative methods work group about 3 or 4 years ago under the Office of the Chief Scientist, Office of the Commissioner. I chair that with Donna, so it's a regulator and a researcher chairing it. It includes very senior members from all of our program offices. And we discuss as an agency alternative activities. We provide representation to other committees, and we're not doing guidance, but we are very informational. So next slide. And we have a website here. This is our webpage. If you go to Google and you type in FDA alternatives, you'll come to our website. And this is essentially a page for the Alternative Methods Work Group to share information to our stakeholders. What's happening right now is that the Office of the Chief Scientist is updating this to be a much more informative website on what we're doing with alternatives. So if you check back periodically, you'll see a lot of new things on this. And one of the things that you do, and one of the things I do when I look at other web pages and stuff, is look for partnerships. We don't have big research labs. We have a lot more regulatory questions in research labs. So we're always looking for, especially in the food area, partners like you guys, yourselves, to work on joint research that might answer our regulatory questions. Next slide. On our website are all of our papers that have an FDA author on them for any types of in silico, in vitro models, alternative organisms, etc. And so this is a library of our publications co-authored by FDA on alternative methods. Next slide. One of the things we're doing with the alternative methods is the papers is to make it a little bit more informative. We're adding the abstracts instead of just having a list of papers. We're putting the abstracts in and a link to that paper in order to make it easier for people to look at it instead of just a list of papers. The other thing that's been very successful is the FDA's Office of Chief Scientist webinar series on alternative methods. This is an opportunity for developers to present new methods and methodologies to the US. We've been holding them monthly. We've had about 60 so far. We may have to go to twice a month because we have so many developers that want to participate in this area. And next slide, I have the criteria. Now, because we're a government agency, we always have criteria on things. So what we want to do is if you have a new method or methodology that you have a proposed context of use and it's a mature method, and you have a description of the gap that it could do and maybe any publications, we invite you to present that to FDA. So we would like to be able to help any developer of new methods, including other governments, to help advance it, but we just don't have the resources to help sort of beginning methods. There are other places in government that can do that. We want mature methods, but what happens then is some of these mature methods are adopted then by a particular center to help work, to help advance the technology. My center adopted one that was using organoids to do ADME. We felt that in the food industry, this would be appropriate method. And we have not brought it into our lab, but we talk actually weekly with the developer to test compounds that we wanted to see, that we had concordant animal study on, and help develop that method so it could become a regulatory tool for the FDA's food program. I know that we have partnered with many organ-on-a-chip companies, including and especially Emulate, to bring organs on a chip into our laboratories. And we have the chips, not only Emulate, CN Bio, Tissues, Memetis, into our laboratories, not to develop chips for ourselves, but to really understand, how this method works, how we could qualify it. So they're really learning experiences in our laboratories. Can these chips answer regulatory questions, and how can we qualify them to do that? Next slide, please. This is our alternative report that we put out in January 2021. This gives you, and this is on our website. Again, if you just go to FDA Alternative Methods and go to it, please, because we're looking at how many hits we get. So We gave you a snapshot of what we were doing across the agency, nationally and internationally. So it's a very informative report. One of the things, if we have to do another report that I think should have been added, is we're describing a lot of the research that FDA is doing, but we're not talking about the regulatory question that we use to do the research. And I think that would add a lot of information to our stakeholders about what we're interested in at the US FDA. Next slide. So we do have some, we do already have some qualification programs. We have the FDA tool development program. This is in our medical device area where they qualify medical device developmental tools. They already have a list of qualified tools. And if you go on our website to CDRH, you can see that. Next slide. So the CEDAR, Center for Drug Evaluation and Research, also has a drug development tool qualification program to look at biomarkers, to qualify biomarkers for use in some non-clinical, but also clinical biomarkers. There's a list of qualified biomarkers on our website. This program, DDT, started out with methods and then migrated over to biomarkers. We're going to be using some of the ideas and guidances from this program to look at biomarkers in food and to partner with the UK and Precision Talks on looking at biomarkers for food safety. Next slide. Now they also have the iSTAND program, which is a new program to qualify. It's a pilot process to qualify tools for drug development. Again, you submit an application, there's a 3-step qualification process. And then once that drug development tool is qualified, it becomes public, okay? There's a public meeting, a white paper, guidance, etc. It's a very transparent process. Once it's qualified, any stakeholder can use these tools in the development stage. One of the things to really emphasize, though, is that for any application for a product at FDA, you don't have to use a tool that has come through a qualification program. In fact, if you don't want anyone else using your tool, you can submit the tool and the underlying data to the FDA as part of the application. And any really senior regulatory person, especially, will be able to look at that underlying data to have confidence in what you use the tool for. So you don't necessarily have to make what you're working on a public for other people to use, and you don't have to qualify it in order to use an alternative method in the regulatory application at FDA. So next slide. Well, we also have ICH, which you know about, and BICH, which is the International Cooperation and Harmonization of Technical Requirements for human medical products. And this is one for veterinary medical products. And you see it's got a lot of members worldwide. ICH started, the human one started, on which BICH is based, started with the European Union, EMA, Japan, and the US, and has blown out into everything. Now, this is not, they do have some guidance, especially in ICH, for how you use alternatives, but the real beauty of these programs is that if you have the same requirements and protocols for animal studies in all the countries globally, you're not repeating these studies with different protocols as you put in applications across the globe. And that saves a lot of animals because you're just doing one study and it's approved across the globe. So next slide, this is going to have a couple of little, yeah, there, okay. These are two people, these are two that are observers and are going to probably become members of the ICH. Next slide. Up, another little leap here. Next slide. Okay, what's probably interesting to all of you is what we formed with EFSA called the International Liaison Group on Methods for Risk Assessment of Chemicals in Food, or ILMREC. We created this through a partnership with the FDA Foods Program and EFSA to facilitate and coordinate the research and regulatory efforts on NAMs through an international framework that will provide the best path towards harmonized outcome. We also created a work group on new approach methods that is new, but we've already selected the first list of priority actions for increasing the use of NAMs. EFSA put out their roadmap on NAMs. We know that the UK is also putting out a roadmap on NAMs. And we have commented on the roadmap on NAMs and agree with it here in the US. Next slide. Next slide. We have technical, oh, no, that's not it. We need the next slide.
So, sorry.
Okay. The next slide has China on it too, actually. Okay, here is the, I think China's a member of this too. Yes, the Chinese National Center for Food Safety Risk Assessment is part of the ILMARAC working group on new approach methods. So we have a lot of people working together, experts from a lot of ILMARAC organizations, and we also invite non-ILMARAC organizations to talk to us on specific topics. We just had a meeting last week in Singapore on nano and new approach methods that was put together by EFSA and Singapore. And we are now inviting Singapore to join the ILMARAC and the work group on new approach methods. And we have one of Dr. Tarzana co-chairs this. He also left EFSA to work with PARC, which is the regulatory program in the EU, but he's still going to chair this committee with me. And you can see we have a diverse number of people from across the agency in the food part. So next slide. And we have priorities. So even though we're a new group, we already have priorities on, we've had some really exciting methods. Our first challenge is to cover the key data gaps with NAMs. We want to try to minimize the request for additional in vivo studies. One of the things that we identified was mixtures. You know, mixtures in food are a problem. In the US, we don't look at mixtures. We look at one commodity, one chemical and one commodity, but that's not what people are exposed to. And we know that we don't think we can solve, or we know we can't solve this problem by more animal studies. We feel that we can develop tools to look at mixtures. And even though the mixtures may be different globally, the tools to assess them could be harmonized. We want to provide guidance for using non-guideline our studies, including harmonized reporting, and to explore the context of use qualification approach, which I talked about earlier in looking at validation. And we want to really create an evolution or revolution in the risk assessment paradigm, facilitating the integration of mechanistic information rather than apical endpoints at the end of a study. Apical endpoints are usually what you use at the end of an animal study. Mechanistic information would include AOPs and IATAs. I'm very excited about what the European Risk Hunter program is doing on developing quantitative AOPs, because then I think it will make them a more appropriate regulatory tool if we know potency at each of the key events, from the molecular initiating event to the adverse outcome. We know how it goes back and forth according to the exposure. So that's a really key thing that FDA is going to get involved in with Risk Hunter, which is one of the programs in Europe, to try to push these forward to use more mechanistic data rather than animal data, to use more biological relevance in qualifying or validating a method. Some of these concepts are going to be included in the ICFAM. ICFAM is the Interagency Coordinated Committee on Validation of Alternative Methods, and it's across 17 agencies in the US, and we are redoing our guidance on qualification to include some of these very points. And we're looking for case studies, and certainly we're looking for partners in your country and other places to test some of these case studies so that we can see if some of our ideas are really feasible and how we go about it, and to give guidance to other people. Next slide, please. So we've had some really exciting talks. This was held in June of 2022, assessing mixtures we included FDA, EFSA, BFRD. We brought in people from other areas and really talked about what we were going to do with mixtures, how we could scale it, etc. And this is an ongoing topic, I know, in the US and probably in your country too. So next slide. The other thing, and I think the most important and the biggest stumbling block for new approach methods to be used as regulatory tools is how are they relevant to humans? So we're looking at human relevant scenarios, lifetime exposure, susceptible populations, gender differences, disease status. We want to consider human relevant characteristics and scenarios. We want to integrate exposing and biomonitoring data into the risk assessment. And we want to decide how we can use human-based advanced in vitro models that represent subgroup characteristics in these. So, and develop criteria for assessing human relevance without using animals, or at least maybe using legacy animals or very targeted animal studies. And this relevance issue is a big debate across FDA, including looking at organs on a chip that use human stem cells. At some parts of our agency want to develop animal chips with animal studies on it to validate human chips, etc. That's a debate. Where do studies come in? We're recently talked to Hopkins about looking at virtual control groups using animal data. So we are exploring this both within FDA and globally on this relevance issue. And we hope to involve all of you guys, if you want to, in some of these discussions. Next slide. And these are other areas that we agreed on to talk about in ILMA-RAC: organs on a chip, cluster of relevant toxicology endpoint proposals, promising tools. I started the organ on chip program when I was in the office of the commissioner. Right now in FDA, it's emphasized for pharmaceuticals, but I really feel strongly that organs on a chip and microfluidic models, so not only on the chips, but organoids, can be used in the food area. And we need to really explore that. We're talking about having a meeting, conference chaired by FDA and IFM on looking at how can we use organoids, organs on a chip, more widely in chemical risk assessment. And there's a MPS summit that's going to be in Berlin next year in June, and we hope that's going to be a topic on hunting. We also want to look at other key areas: developmental neuro, non-genotoxic carcinogenicity, immunotoxicity, endocrine disruptors. All of these are things that you're looking at too. We recently reviewed a IATA from Syngenta using a pesticide and looking at carcinogenicity without a rodent study. That was really exciting. We are inviting OECD in to talk about the IATA for developmental neuro. Is that something that we could use? Developmental neurotoxicity is a big area for us, especially in children's food. We have a Closer to Zero program that looks at all these metals that we see in children's food that are either labeled for infants and children or commonly used for infants and children. That's a big program. Developmental neuro is the endpoint that we're looking at, and we're looking at really better ways to assess that. Next slide. So you see that under ILMA-RAC, we'll be very busy. And the other things that we want to talk about under ILMA-RAC are investigating the use of AI in food safety NAMs to identify data gaps and reduce animal testing. Looking at AI-based technologies, discussing the requirements for proper high-quality data collection, discuss privacy and confidentiality issues. We want to look at neurodegeneration And we want to look at chemical produced during food processing. So one of the things that's a big topic for us that we know we have to use in the food area, we know that we have to use new approach methods are these heat process contaminants. There's so many of them. And I'm talking about acrylamide, furans, 3-MCPD, 4-MEI, all of those that are produced during the heating or processing of food. It's essential to process food or you're going to have microbiological hazards, but these toxins come up. They're not considered food additives, so they don't come under the Delaney clause in the US. But how can we look at them and what can we do about them? We can't do animal studies. There were so many animal studies done on acrylamide, and I don't think anyone can afford to repeat that. That's one of the reasons we partnered with a company that's doing using organoids to do ADME, and we are putting through a couple of heat process contaminants to see that we already have animal data on to see the concordance of this new model. Next slide. And you can see where ILMARAC's going to be very busy. We want to do a guidance on metabolism, integrate different NAM approaches into a guidance on how it can be used. We want to look at nanomaterials and nanoplastics. Nanoplastics are being found in food. How are they getting there? The other thing we're looking at, I don't think that's here, is tattoo pigments. Tattoo pigments in the US are considered cosmetics. And we are looking at how we know that they become systemic almost within the time they're put on your body. They're not made for humans. We know they become systemic. Do they cross the placenta? And in essence, tattoo the fetus. So this is another list of things we're looking at, and we need all the partners that we can get. And we certainly encourage any of you that if you have case studies in any of these areas or want to come to some of our meetings to please come. Next slide. So another thing that we're doing at FDA is we have a new alternative method program across the agency if we get additional funding to expand the qualification process. And each of our program offices, as I told you, there are 6 of them. I described 3 of them have qualification programs, but Foods is the CIFSQ Foods program is developing a qualification program too. And in the last slide, last speaker, their definition of NAMs, I really like, because that's the first thing we're going to do is start with our definition of NAMs and then look at where the food industry is. What is the food industry using in the US and internationally for new approach methods? What new approach methods would they use? And how can we work with them together? Because it's no use. We found even if we would qualify methods, or we can't have in the US, we can't sort of mandate developers to use new approach methods in lieu of animal studies. We can suggest it, we can recommend it, but we can't mandate it as you can in other countries. So this is another collaboration to fill in the gaps, to develop more policy and guidance on an agency basis. And next slide. So we also have gone to the FDA Science Board to ask for input on how the agency can enhance its existing approaches to support the development of qualification and implementation of alternative methods to reduce, replace, or refine animal studies and to improve predictivity of non-clinical testing. So last slide. So I hope I've given you a snapshot of what's going on in the US and globally in food safety. I hope you're encouraged to join us, at least with case studies, on how we're trying to, as a global family, bring new approach methods into the food safety risk assessment paradigm. And to remember that change takes time, but it will happen if we work together. So thank you very much for allowing me to speak today. I look forward to hearing from you. We do have you have my email, it's on the next slide. We have also an email for our website, alternatives@fda.hhs.gov. please let us know of any collaborations. We encourage you to work with us. You can see China is already part of ILMERAC. And I think as a global community, we can really move this forward. So thank you very much. And I'll answer any questions.
Thank you, Sue. Nice presentation. So any questions or comments? 大家有问题吗？
Yeah.
Any question?
I have a question. And Professor Fitzpatrick, we are talking about the alternative methods in the field of food safety evaluation today. But my questions maybe it's not for the food safety evaluations. We may notice that several days ago, the U.S. Senate passed a bill to revoke animal testing requirement for drug developer. I'd like to know, is there any kind of detailed strategies or plan for, any kind of regulatory guideline for supporting the applications of pharmaceutical products?
So can you repeat the question? You're talking about what's in protein products and drug?
Yeah, I mean, we know last month, at the end of last month, the Senate passed a bill to revoke animal testing requirement for drug developer. I'd like to know, is there any kind of plan or detailed plan or for, the publish, figure out any kind of regulatory guideline supporting the R&D of the pharmaceutical products?
Well, I know that most CEDAR works through ICH, and they've published some guidance on alternatives, maybe not in the area that you're working in. If you have,
I mentioned the FDA modernization 2.0.
Yeah, okay, that's it. I know that they have encouraged us. The 2.0 said they're not saying we can no longer use animals. They can say that says that we have to move towards non-animals. So
Yes.
Right now we don't have the same restrictions that in other countries about using animals. I'm sure it's coming, but and I know that they criticize our laboratory in Arkansas for having a budget for animal studies. That's why we're working so hard not to use animals. But again, just because we say in CEDAR, for example, we would accept organ on a chip data, we're not seeing it because we can't make people use alternatives in regulatory products. We can only suggest it. So right now, even though we put a lot of time and energy, we work with the IQ Consortium for MPS. We've worked on chips for since 2011. We're not seeing any data in a regulatory application.
So thank you. Maybe we can contact with you and for more, discussions. Thank you.
That's fine. Yes. Why don't you email me? And,
Yeah, I know your email address and.
And we can talk about if you have proposals too, we can present them to FDA, to CEDAR. I mean, I work with them all the time. They're a part of our alternatives committee. So
thank you.
Any other questions?
Yeah, and I have one more question.
Okay.
This is Santiago from the Center of Disease Control and Prevention. So thank you so much for your nice presentation. My question is, we have two lamps that NAMS. One is a new approach methodology. Another is, you said, is new alternate methods. So can you tell what's your point on these two different lamps? Thank you.
So you understand the question?
No other questions or?
The difference between new approach methodology and the new animal testing.
贾教授他可能我估计听不见，可能好吧。
So can you hear me? So, Dr. Fitzpatrick, can you hear me? 可能听不到线上，可能听不到线上。嘉宾，
那贾老师算了算了，
啊，那就
没事
OK，那就算了啊，可能听不到，听不到
Okay,
咱们线
that's all. Because of Professor Fitzpatrick can't hear the question from online
No, I can't hear.
audience.
If you have questions, you can email me too, if you don't want to. In English, though.
Methodology. The second name is non-animal method. So the difference, could you share your opinion with us?
What's my opinion?
Yeah,
My opinion on,
the two names for the question from Dr. Guo.
okay, on new approach methods. Yeah, I can't really, I can't hear all the questions. You want my opinion on certain NAMs?
Yeah, he means you know.
I mean, ordinarily, I mean, FDA wouldn't give an opinion, which takes back like over in a meeting. I mean, I can write, I can talk to you about it, but I would, just hearing something, I can't, I hope it's a good NAM, but I don't know.
Okay, okay. Thank you.
I'm sorry, yeah.
Okay, 嘉宾。
So, okay, so that's it.
好，谢谢。
OK OK，好，还有其他的问题吗？这样的话，因为时间的关系
Any other question?
非常感谢。
Yeah,
Okay,
any other
for the interest of time.
Yeah, please, just if you have questions, just email me. Think about joining the NormAccurate. And remember, we're all partners in this like global NAMS adventure. So thank you. I'll sign off now. So, bye-bye.
Thank you。彭老师，你看现场还有问题吗？
没有了。
好，
No question.
大家哈，
Thank you for
Thank you.
staying with us.
线上线下
And many thanks to the audience online, offline, and okay,
Bye-bye.
that's all for our
Thank you all
session
for
G.
international speakers.
Mm.
Bye bye.